Questo almeno. Mm, aynen. Aynen. İşte işte. Hiç kimse yok onlarla. Non mi ricordo neanche.
Farmers all around. And generation time. It takes typically 5 months or so to go from seed to seed. And that's not a very good thing if you're a PhD student working on wheat. It's not like Drosophila or yeast, for that sake, where you can do lots of genetics very fast because of the short generation time. So confronted with that problem, Lee Hickey, a researcher in the University of Queensland, he came up with a process called speed breeding. And we worked with Lee Hickey to perfect this process. The plant on the left is about 5.5 weeks old. It's grown under our standard conditions in the John Innes Centre, where I used to work. This plant here on the right is also 5.5 weeks old, and it's grown under speed breeding conditions. And under these conditions, we can go from seed to seed in just 8 weeks, 2 months. So what is the trick? We have done away with the old sodium vapor lamps that produce a lot of light in the green and the yellow part of the spectrum that you can see up in the right-hand corner. And that is not very useful for photosynthesis. Photosynthesis works better in the blue and the red part of the spectrum. So we replaced the old sodium vapor lamps with LED lights optimized for blue and red light. And then we increased the light intensity, and we kept the lights on for 22 hours to trick the plants into getting very little sleep. And under these conditions, we were able to cycle the plants very fast in just 8 weeks from seed to seed. And it works not just for wheat, but also for barley and many other crops and their model derivatives. The other challenge of working with wheat is that the genome is very large. It's about 150 times or so bigger than the Arabidopsis thaliana genome, the lab rat of the plant world. And there's a lot of genes in wheat compared to Arabidopsis. And within these 120,000 genes, we have the immune receptors, the genes encoding disease resistance. Now, it turns out that most disease resistance genes that have been defined by genetics and subsequently cloned in plants over the last 30 years, they encode a type of immune receptor known as a nucleotide binding site leucine-rich repeat. And armed with that knowledge, you can compare these genes with each other. You can compare the sequences, and we call them NLRs for short. So you can compare NLRs, and there are certain motifs that are represented again and again. And these motifs are numbered up here. And so if you take those motifs and scan a genome, you can very quickly annotate, you can find the NLRs in the genome. And there's about 3,400 NLRs in the wheat genome. So the genome is using a lot of its potential to encode for these genes. It really cares about immune receptors. Now, traditionally, genes have been cloned in wheat using map-based cloning, so using genetics. This is, it's pretty slow, and you rely on recombination to get close to your gene. And so some years after starting this journey, we introduced mutational genomics into wheat. And what we did was we would take a wheat strain where we had defined by genetics that there was a single disease resistance gene against a certain isolate of the pathogen that we were working with. And so we would take a big bag of this seed and mutate it with a chemical mutagen, typically EMS, grow up the mutant population, infect it with the pathogen, and then let the pathogen do the work to identify susceptible mutants. And then we would use enrichment sequencing. We would use exome capture to sequence just that small proportion of the genome encoding the NLRs. And this works very well. We could design RNA probes that are complementary to all the NLRs and then fish out sequences corresponding to these NLRs and then use next generation sequencing or Illumina sequencing to sequence the NLR-ohm. And this worked very well. We got about a 1,000-fold enrichment. The other way that we could reduce the complexity of the wheat genome was to collaborate with Jaroslav Dolezal in the Czech Republic. At the time, he had fine-tuned the art of chromosome flow sorting in wheat so that he could get pure preps of single chromosomes. In the diploid, that would reduce the complexity 7-fold. In hexaploid wheat, that would reduce the complexity 21-fold. And so what we would do, we would sequence the resistant parent, align all the 3,400 candidate immune receptors, and then we would sequence a handful of mutants. And we would line them all up, and we would look for all the NLRs that have mutations in them. And we would, in particular, look for an NLR which would be mutated in all of the independently derived mutants. And the probability of that occurring by chance alone is very low. So that would be our candidate disease resistance gene. And in fact, that worked very well. There's some publications on the right where we reduced this concept to practice. And the one at the bottom, I like to point to, where we cloned YR5 and YR7, that was work done by a PhD student in the lab, Clémence Marchal, and she managed to clone 2 disease resistance genes within the confines of her PhD, which at the time was quite a remarkable feat. Now, some of you will notice that some of the genes we cloned were for stem rust. Stem rust is a devastating disease of wheat, which can cause epidemics and destroy the crop. And historically, it's been associated with crop failure and famine. And for many years, scientists and farmers have realized that stem rust has an alternate host, which is the Barbary bush. And it requires the Barbary bush to go through sexual reproduction. And so for many years, farmers and communities around the world have tried to destroy the Barbary bush. This is a campaign poster from the early 20th century in the US, where they were encouraging farmers and communities to destroy the Barbary bush. And they would do this by pouring salt onto the plant or going out and using horses to yank the plants out of the soil. Here's a more sinister campaign poster, which came in the wake of the Bolshevik Revolution, where the Barbary or the stem rust was likened to the red-handed anarchist of the grain fields. Going further back in history, ancient farmers in Greece and in Rome, they had a god called Robigus, and they would sacrifice a red-haired animal every spring to pacify Robigus and protect their crops. And this is our first paper that we published on cloning stem rust resistance genes in wheat. And as you can see, the Robigalia Festival was held on the 25th of April, and we published our paper on the 25th of April. Now, don't tell me you don't believe in Robigus now. Some communities in Italy, they still practice this. I'm sure you can think of a fiery red-haired politician to replace instead of the poor dog, but we'll leave it at that thought experiment. Don't go any further. Okay, so we tried to see if we could adapt this method of mutational genomics to the wild relatives of wheat. And here we have an experiment that's ongoing to generate a mutant population of a wild grass called Aegilops sharonensis. You would starve to death if you depended on sharonensis for your daily bread, but it has remarkably good disease resistance. So we started this project. We had to do a lot of so-called breeding in sharonensis to genetically isolate a single stem rust resistance gene. Then we had to bulk up the population. And then we had to harvest several thousand seed, 20,000 seed, and we had to peel them. It's very difficult. It's so difficult. It's almost like putting bamboo splinters under your nails, a horrible process. Then we mutated the seed, and about 6,000 of the seed survived the mutagenesis, and they grew very well in this glasshouse. We took up the whole glasshouse in the John Innes Center, but not a single one of the plants were fertile. None of them set seed. That was very disappointing. And in fact, it's very difficult to mutate a diploid because there's no genetic redundancy in the housekeeping genes. This is a lot easier in hexaploid wheat, where you have 2 or 3 copies of all the housekeeping genes. You can hit them really hard with mutagenesis. So we had to give up this experiment, and we realized that we would need different approach. We would need to do genetics or create populations that would not require laboratory controlled genetic structuring. And so we decided that we would go and use the hand that Mother Nature had already given to us. And that's represented up here in the left-hand corner. This is the genetic structure of another wild wheat, the one I introduced in the earlier slide. This is Aegilops tauschii, the D genome progenitor. And here we can see the natural genetic diversity that has been generated over hundreds of thousands of years by natural recombination in the field and natural spontaneous mutagenesis. And so we reckoned that if we would phenotype all the individuals in this genetic structure with a single isolate of the pathogen, we would get a range of phenotypes. And then we reckoned that if we used our resistance gene enrichment sequencing, we could sequence the NLR-OME or complement of all of these individuals. And then we would be able to do association genetics to derive correlations between phenotype and genotype, and hopefully identify candidate disease resistance genes. So to reduce that concept to practice, we decided to work on this genetic structure, and we did resistance gene enrichment sequencing on 150 accessions of Aegilops tauschii. And then we did association genetics for disease resistance. Our first trial was with a isolate or a race of the pathogen called RKQQC. This was a positive control because this isolate of the pathogen is diagnostic for a disease resistance gene called SR33, which had already been cloned after introgressing into wheat. So this was very useful because we could test our method. And lo and behold, we got a single peak in our genome-wide association plot, which corresponded to SR33. So this was very exciting. But of course, you can, when you're doing bioinformatics, it's easy sometimes to find what you know you're looking for. So we had to do it with the without positive controls. And so here's another one with a different isolate of the pathogen called TPMKC. And here we were able to identify SR45, which is actually another positive control, but we were also able to identify a peak which genetically corresponded to another gene that hadn't been cloned called SR46. And we were later able to confirm that this candidate gene was indeed SR46. And finally, with QTHJC, we were able to identify a candidate gene for SRTA 1662, and we were later able to confirm that this was indeed the right gene. So in conclusion, with a bit of sequencing and phenotyping and association genetics, so bioinformatics, we were able to identify all the 4 major stem rust resistance genes that had been genetically defined in this species over the last 40 years. Okay, so this was all very well. This allowed us to clone disease resistance genes encoding NLRs. But we really wanted to be able to clone genes that were not encoded by NLRs and be completely unbiased in our approach. So therefore, we decided we had to sequence the whole genome of every member in the panel. And this was something I proposed quite some years ago now. And back then, it was considered a very large task to sequence so many individuals with such a big genome. And so we had to form an international consortium of crazy people like myself working on these wild relatives. And with this consortium, we were able to sequence not just the original panel, but enlarge it to 242 accessions. And with that, we were able to do association genetics for phenotypes other than disease resistance. And so here we've looked at variation in trichome density on the leaves. We've looked at spikelet number, and we've also looked at a pest wheat curl mite, and we looked at a classical disease, powdery mildew. And interestingly, for powdery mildew, we got, as you can see, a very discrete peak on chromosome 7. And under this peak, there were no NLRs, but there was a wheat tandem kinase. And we've later confirmed that this wheat tandem kinase is indeed the disease resistance that confers this resistance to powdery mildew. So that was very gratifying because we really demonstrated that now we could clone disease resistance genes that are not NLRs. And that was published a few years ago in Nature Biotechnology, and it was a very large consortium effort. Now, recently I moved from the UK to KAUST in Saudi Arabia. And with generous funding from KAUST and also with support from the Open Wild Wheat Consortium, we expanded the size of the panel to nearly 500 accessions. And we also generated some very high quality assemblies using long read PacBio sequencing for 46 of these accessions. And then now we are working with the community to look at lots of different traits that are of strategic interest. And we continued our interest in disease resistance. And with these new resources, we were able to clone a leaf rust resistance gene called LR39, which also encodes a wheat tandem kinase. And we were able to finally give SRTA 1662 a gene designation, SR66. And that was published just last year in Nature, and again, another large consortium effort. And so now it's like being a little child again and going into the sweet shop. We've cloned a lot of genes. And what do we now do with all those disease resistance genes? We're spoilt for choice. So we have collaborated with Mick Aliff and his postdoc Ming Lu in the CSIRO in Australia. And they have taken some of the genes for stem rust that were cloned by us or other members of the community, and they have generated a stack called the Big Five. And as you can see on Mick Aliff, you've got to be really strong and big to make these stacks. And this was transformed into a cultivar of wheat fielder, which is very susceptible to stem rust. And then it was tested in the field in Rosemont, in Minnesota, in the States, in the US, by Brian Stephenson and his postdoc, Oadi Matni. And as you can see, the non-GM is completely susceptible to stem rust in the field, whereas the Big Five is completely immune. And Brian, who has worked for more than 30 years on stem rust, he said he had never seen anything like it. So this was a very, very gratifying and exciting moment. Now, there is a bit of an elephant in the room, and that is that this is a GM product. And there's a lot of socio-political opposition to GM wheat. And in an opinion piece that I published a few years ago with Kanwa Palduga, we likened GM wheat to a GM orphan crop because a lot of our major crops like cotton has BT, maize has BT, and soy is also GM, canola is GM, but wheat is a GM orphan crop. But I would say there is light at the end of the tunnel. There's a small biotech company, largely unheard of, in Argentina, and they've been working on the quiet for nearly 20 years to develop a GM product in wheat. They have introduced a gene from sunflower which confers drought tolerance. It increases the yield in low input environments by about 16%. And recently, they managed to deregulate the so-called HB4 wheat. So this is very exciting. And lots of jurisdictions, they are now accepting HB4 wheat. And importantly, in 2021, HB4 wheat was accepted by the authorities in Brazil. And that was a really important watershed moment in GM wheat because Argentina exports a lot of wheat to Brazil, and that makes this GM crop commercially viable. And now jurisdictions around the world are jumping on the bandwagon and they are deregulating HB4 wheat. And I would argue once we have a GM crop, it will be easier. It will lower the barrier to introduce additional GM traits, such as for disease resistance. And so we are working with Bioceres in Argentina, and they do care about, this is a picture of HB4 wheat on the right and the control on the left, grown under drought-prone conditions. And as you can see, the HB4 wheat grows very well. But it does suffer from stem rust. So we're now trying to introduce a GM stack for stem rust into the HB4 background. But it is not so straightforward politically. A lot of these genes that have been cloned have been protected by patents. And you can see there are two main players. There's the CSIRO in Australia. They've cloned a lot of genes and they have protected these with patents. And for many years, I was funded by the Two Blades Foundation. And they have also patented most of the genes that I cloned. So that complicates your freedom to operate. And you can see that many of these genes, patents, they will still last for very many years. But the landscape is rather checkered, whereas jurisdictions like Australia, the USA, and the European arena, they have the patents, they cover these genes. In South America, you can use most of these genes without any restriction. So that opens a possibility to introduce this technology into South America without being encumbered by patents. And so now to some predictions for the future. I think over the next 5 years or so, we're going to see increased activity in gene cloning in wheat. I predict that we will clone another 200 disease resistance genes for rusts alone. I think we will identify the corresponding effectors that these immune receptors recognize in the pathogen population. This will allow us to study the efficacy of all the cloned genes very carefully. And that is important because when we understand the efficacy, we can design judiciously stacks that are going to be more durable. And I think it won't be long before we can then generate a triple rust stack, so that we would create a wheat that would not have stem rust, stripe rust, or leaf rust. And I think this will likely happen as part of a public-private partnership, but it is a complicated exercise which will require a lot of diplomacy and a lot of politics. And so one of my final conclusions is that when you work on wheat, there's a little bit of science, maybe about 20% science and 80% politics. And so finally, I'd like to thank some of the many funders who have supported me over the years. I could still need more funding. So one day I'm going to steal the handbag from an old lady crossing the road, an old rich lady. That's what I was thinking about anyway. But instead, I went to Saudi Arabia and I'm now using money from a dead man, a very visionary individual, King Abdullah, who sadly passed away some years ago. He was very visionary. He funded or founded the King Abdullah University of Science and Technology. It's a very well-funded institution with researchers from all over the world. There's about 100 different nationalities in KAUST. And it's really a lovely place to do science. And this is the view from my office, by the way. No, I'm joking. This is my group. I'd like to thank my new group in KAUST. And that's it. Thank you very much for your attention.
Thanks so much, Brandy, for the entertaining, wonderful, exciting talk. And I expect that will be a lot of questions here. And who want to start? Or are there any? Yeah.
Thank you very much, Brandy. Eclopes tavşan is how it calls? Ajda adı mıydı? Ancestor of bread wheat, yes. So if we do GWAS in Eclopes tavşan, it's work in bread wheat or not? Sometimes.
So what is the question? We can do
Yeah.
genome-wide association
Yes.
studies in
Yeah.
Kaohsiung.
Yes.
We can identify candidate genes. And then the question is, do those genes work in wheat?
Yeah.
We can incorporate the genes into wheat by wide crossing, and we can make synthetic hexaploids or direct crosses with Taoxiai and introduce the genes. So you can do it in a natural way that is exempt from any regulation. And I think most of the time the genes, they do work in the hexaploid background. Sometimes you find that they don't work. And that might be due to dilution when you put it into the hexaploid background. And so maybe you then need to find a way to overexpress that gene or screen many individuals to find one where that expression works. The percentage, I don't know. We test most of our genes by transgenesis, and most of the time that works. But we do find occasions where it doesn't work as well in the hexaploid background. And I will, in my next presentation, I'll give an example. I'll get close to an example of that. Some resistance genes are digenic, and they require two factors. They require a sensor factor that can perceive the presence of the pathogen, and they require a helper factor that can signal downstream resistance. And if they're not genetically linked and you just introduce one of them, you may leave behind the other one, and then the gene doesn't work. So sometimes you need two genes.
Thank you. Good presentation.
Are there any questions?
Hi, Brenda. Great science and a nice delivery as usual. Hopefully you have cloned many genes genetically, and we hope that these great wealth of genetic data will be transferred into practical breeding soon. So the main question is, my question is now, so you have cloned many genes, including the gene for curl mite. So do you have a co-segregating markers for simple mass system?
Yeah, so we haven't formally cloned the gene for curl mite yet. We have used GWAS to define a genetic region which by definition should contain the gene. And this is not our area, someone else is following up on that. Generating markers we have for some of the genes we've cloned, like the YR genes, the YR5, YR7. As part of that publication, we generated genetic markers. We've also generated genetic markers for some of the blast resistance genes that we cloned in collaboration with Paul Nicholson. But it should be straightforward to take the sequences of the genes that we've cloned and use those to generate perfect markers. And yeah, the sequences are published. Anyone can do that. Thank you.
Thank you very much, Dr. Brandy. So my question, we know a lot of resistant genes cloned and identified, and some of them not cloned yet. Did you focus to clone some newly identified genes nowadays?
Yes, so some of the genes that we've discovered in the wild relatives are so-called new. They haven't been genetically defined and designated. That's one of the advantages of working on some of the more wacky stuff like Aegilops sharonensis. I have a love-hate relationship with sharonensis. It is so difficult to grow. The agronomy is so poor. It has all the hallmarks of being undomesticated. It has extraordinary genetic diversity for disease resistance, and very little of this genetic diversity has been introduced into the wheat breeding pool. And so we hypothesized that if we focus on some of these wild relatives that haven't been used in breeding, we may discover new broad spectrum disease resistance genes that have not been, let's say, exploited by the breeders and overcome through deploying them as singletons and selecting for variants in the pathogen population that are virulent.
Okay, so from the questions from biotechnology, recently we had a colloquium in our university. It was a talk about NLR engineering, and what do you think about the future of NLR engineering? Would it be possible to do really engineering of NLRs for resistance?
Yes, there are several emerging examples of being able to engineer NLRs to widen the spectrum of recognition or indeed recognize altogether new effectors. And for that to be successful, you really need you need the 3D structure of the NLR and the effector and any other signaling components that work in concert with the NLR. So I think the future is bright. And if I were Demrust now, I would be shaking in my spore.
Great talk. So coming from the effector side, so do you think that the effectors are engaging directly with your kinases, or is it an indirect effect? I mean, do you have any examples for how that works?
Yeah, in fact, I have a slide on that in my next talk. But in some cases, the interaction is direct between the receptor and the effector. And in many cases, it's indirect. And I think one of the most seminal contributions in this field was made in 1998 by a postdoc in the group of Jonathan Jones, Eric van der Biesen, who hypothesized that in many cases, the interaction is indirect, and that in fact, the effector is targeting a pathogenicity target in the host, and the NLR is guarding that pathogenicity target and can detect the effect of the effector on the pathogenicity target. And so it's an indirect interaction. And this is a beautiful way of explaining how a host can defend itself against a myriad of effectors with just a few thousand, or in many cases, just a few hundred receptors, because there's a finite number of cellular hubs that are targeted by effectors. Jochen.
Randa, thank you very much. It's a really great talk. And in particular, I like your visionary style. And I have two questions. Second questions will be somehow visionary. You talked about these receptors, but when you showed the survey of cloned genes, you also touched on, without commenting on, durable resistances, LR34, LR67. Wouldn't it be a good strategy to stack them in these panels of resistance genes?
Yes, the so-called APRs, adult plant
durable
yes.
resistances, which rely on entirely different mechanisms like transporters.
Yes, completely. In fact, one of the genes in this big five is one of these broad spectrum resistance genes. It was a little hidden. Because we used in that slide, I used a different designation. I think it's LR34, which is also known as SR55 or so, because it confers triple rust resistance, but that is in the stack. And now the latest thing is that Mick Aliff he's got his big five. He's made a new stack called, I think it's Fearless 5. And if you combine Big 5 and Fearless 5, you have 10 stem rust resistance genes. I think this is going to be a very tough nut to crack for the pathogen.
Absolutely, yeah. It is in part answers my second question. So the breeders community is a quite nice community, and they think a lot together. But when it comes to individual success, they are egoistic. And if a breeder has a successful cultivar with just one resistance, which belongs to your stack, then this may be overcome, and another breeder has a second, and so on. Yeah. And of course, even though you would have 5, there is a risk that these will be overcome. But of course, 10 is another league. Yeah.
Absolutely. That's you put your finger on it. I think we need to clone more disease resistance genes from these wild, wispy, insignificant looking grasses that the breeders, they wouldn't even touch. There's a lot of genetic diversity out there. And the technology for cloning these disease resistance genes is improving. We started off driving a Ford Fiesta 15 years ago. We're now driving a Ferrari. And I think we're about to step into a spaceship. And so I really believe that in 5 years or so, we'll have another 200 resistance genes cloned, including their corresponding effectors. And then we can study on a worldwide scale the efficacy of these genes. And I think we'd be able to design some stacks that are going to be so difficult for the pathogen to overcome. Not saying they will ever be overcome, but we can have more stacks in the background, next generation stacks.
Maybe I have a general question. So is there any pathogens or disease specific to Triticum? If yes, and do you think they have also kind of friendly conversation, and then how do you recognize the host plant, and then how like host plant also can support the pathogen to establish on the plant?
So if there's any pathogen of wheat which is specific only to wheat, well, it takes something like powdery mildew. There's a wheat adapted version that grows more or less specifically on wheat and durum wheat and some of the closely related grasses. So they tend to be quite specialized and adapted, these pathogens. I think maybe turn the question round and ask, is there any grass that doesn't support rust? And in fact there is, and that's rice. The rice is very interesting because there's no rust that grows on rice. And so people have wondered for many, many, many years, what is the nature of this non-host resistance? And so it's difficult to do genetics on non-host resistance. But, you know, maybe now with CRISPR, we saw some wonderful presentations yesterday on CRISPR. And so could we use that technology to remove all the NLRs from rice and see if it would now be susceptible to rust? I think that would be very interesting, and that would allow us to do genetics on rice for non-host resistance and understand what's the basis of that. And maybe learn from that, you know, how could we turn wheat into a non-host?
Thank you. And maybe thank you so much, Brandy.
Thank you.
Lovely discussion. Now we invite our next speaker, Jochen Kumlin. He will be talking about double coupler technology, which is really interesting. Hozirgi dokladda ya'ni gaploid olish texnologiyasi haqida metodlari, qanaqa metodlar double gaploidni olishda juda ham qulay. Chunki biz o'sha gamazigota o'simliklarni olishda biz asosan klassik yo'l bilan o'sha bir 7 yilda chachittirish orqali olamiz. Lekin aynan bu texnologiyani afzallik tarafi sizlar o'sha 1 yilda aynan gamazigotali gaploid o'simlik olishingiz mumkin va albatta bunda o'sha Yo'qin gapiradigan texnologiya metodlar bizda hali ishlab chiqilgani yo'q. Menimcha bu narsa bizda o'sha hamkorlik o'rnatib, o'sha texnologiyani bizga olib kelish bizda imkoniyati bor. Menimcha sizlar uchun ham qiziq bo'ladi degan umiddaman. Stage is yours, please.
Yeah, thank you, Bano, for this nice introduction. So I get used to stand here meanwhile. My today's talk is about haploid technology, which is a very powerful breeding technology and can be combined with all those things the other speakers talk about, because here we have a technology which produces true breeding material in a much shorter period of time from whatever recombinant material. I will explain in detail how it works. Haploid technology actually relies on the formation of haploid plants. And haploid plants are typically can only be obtained from haploid cells. So the cellular origin of haploid plants is always a haploid cell. And these haploid cells mainly occur in the gametophyte. We may all know with the biological background that we have these oscillations of sporophyte and gametophyte, and the gametophyte is the hidden generation in plants which produces the gametes, which then fuse egg cell and sperm cell to produce a zygote, which is the originator cell of a new individuum. And haploid gametophytic cells we have on the male side, which is the pollen, and on the female side, which is the embryo sac. And what you can see here at the left-hand side is cultivated immature pollen. They are extracted at the unicellular stage. They are called then microspores. So these are the direct product of meiotic recombination. And at the female side in the center of the slide, you see a gynecium, and it is indicated where the ovule resides within the structure, and the embryo sac in yellow and the egg cell in red. And the egg cell, of course, is a haploid cell. But also the progenitor cells of the egg cell are haploid cells and could give rise to plants if we manage. And then there's a third principle of producing haploids. This is a bit complicated, coined uniparental genome elimination. That basically means after fertilization, one of the genomes of the parents is not stable in this plant and is eliminated again, leaving alone the haploid genome of one parent. And this parthenogenesis pathway can be obtained by cultivating ovaries or ovules, or by using particular mutants. And this uniparental genome elimination, dependent on which gender retains as a haploid cell, is called gynogenesis or androgenesis, and can be obtained either by interspecific crosses or by so-called haploidy inducer lines. What all these systems have in common is that you start with a haploid cell. You have to initiate cell proliferation from this haploid cell, which is difficult. For instance, if you imagine pollen, an immature pollen grain has its destination to form a mature pollen, which is typically bicellular or tricellular, or the egg cell actually stacks development as long as it is not fertilized. But by certain stress treatments or other tricks, some people have managed that. So, and once you have cell proliferation, you have a chance that you have totipotent tissue, and this can give rise to plant regeneration. And once you have this plant regeneration, during all this pathway, you need to have one event which is essential for the technology. This is whole genome duplication. Haploid plant is not capable of producing a next generation because it cannot undergo a regular meiosis, a reduction division. And therefore, this whole genome duplication is required to obtain the diploid status again. And as soon as you have this then double haploid plant, you can practically clone this plant because it's entirely homozygous. It will not segregate anymore. And the purpose for the breeder or the researcher is that what you see in this plant is what you get in the next generation. And of course, this is a breeder's dream, not to have any segregation anymore after selecting something which is useful. So now, giving you the first example, this is regeneration from the origin of immature pollen. And what you see here is a panel of developmental stages of pollen development, starting with a microspore just released from the tetrate, which is the end product of meiosis. So the microspore actually is one of these cells of the tetrate. And this undergoes a certain cellular development. It first produces a large vacuole to store nutrients for the later energy-consuming process of pollen tube formation and fertilization. And it undergoes pollen mitosis 1, so a first pollen cell division. And this gives rise to two different cells, the generative cell and the vegetative cell. And the generative cell you see in the fourth picture at the bottom, which is separated at the bottom. And the vegetative cell looks very much like the former microspore. So, and right after this cell division, these two cells move apart from one another in this bicellular pollen grain. And this development goes on, but for us, for haploid technology, there's a certain window of development which is essential for us because there is a certain totipotency in these cells, and these cells can be triggered not to follow the pollen developmental pathway, but cell proliferation and maybe formation of plants. So, and this can be triggered, for instance, by putting these microspores in a medium which lacks certain nutrients, so we expose them to starvation, or we treat them with physical stress like extreme cold or extreme heat temperature. It's a border with what they can survive. So, and what you see here is the picture from the former slide where you have these microspores which have already undergone intense cell division. And when this development goes on, we get kind of microcalli. So these cells develop so much that the envelope of the pollen bursts, the exine of the pollen bursts, and then we have kind of microcalli, and these microcalli can further develop. And in the last picture, you see that they can even give rise to plants. And these are either haploid plants or double haploid plants, dependent on whether they have undergone whole genome duplication. And this basic principle, surprisingly, has been described in more than 200 different plant species, even though it does not occur in nature. So this is really surprising that it is possible to manipulate a very highly specific cell through this kind of general program of producing regenerable tissue and giving rise to plants. So, and very importantly, you have to understand the genetics of haploid technology, and this I will explain in the current slide. So here you have a representative of a hybrid plant, different mother, different father were crossed, and these are represented by the two colors of the chromosomes. So there, I took my my beloved workhorse barley with 7 chromosome pairs, and each individual chromosome comes from a different parent, of course. And as you may all know, during meiotic recombination, they exchange fragments. So, and this is random, and this is the first situation where you have random recombination of genetic information between the two parents. And the second step of random recombination of this genetic makeup is the distribution of these recombined single chromosomes to the gametes. And you can see here 4 as representatives. These can be immature pollen grains, but also can be egg cells. So, and this is also the reason why we all look so different, because of this meiotic recombination. There's endless potential of recombination because we have these 2 steps where genetic information from 2 parents is mixed up in an unpredictable way. So, and when we are capable of developing plants from those recombined cells, then it looks like this. And as I told you, we have these plants have to undergo, or on their way, they have to undergo whole genome duplication. And then the magic happens that all in a sudden, we have all these recombinants as diploid individuals, and they are homozygous across their whole genomes. And thereby we can produce populations of recombinants, and which is very important for the breeder, because the breeder produces genetic diversity. But the genetic diversity in the ideal case is entirely true breeding. And when the breeder now selects according to the traits of these individuals something, it can be identically reproduced. And this is actually the trick of the whole technology. And I will give you an example how it could look like. Here we have crossed 2 different barleys. One has these light grains and one has these black grains. And the F1 grains look like this. But when you now grow plants from these F1 grains and use these immature pollen to produce populations of double haploids, you get this. And I was really amazed to see this for the first time in practice. Here we have produced almost 400 different double haploids, and they are all different. But the magic is whatever the grains they produce from one plant, they all look the same. So you can see here 10 examples of grains from these double haploids. They are all different. They combine different shapes, colors, and whatsoever of this grain, but one plant produces one sort of grains, and this is reproducible over generation. So, and having this culture system at our disposal, we were also interested to see the early processes of when the microspore deviates from the normal pollen development towards proliferation and to produce totipotent tissue. And here you see a regular like light microscopic recording. But when we teamed up with Michael Melzer's group, we were capable of following the development microscopically and producing little videos and see what happens. And to further empowering this technology, we used transgenic barley, which carries a GFP gene and accumulates GFP in the nucleus. So we can particularly follow the nucleus. And this looks like this. So actually, it's exactly the same object, once recorded by light microscopy and once recorded by confocal laser scanning microscopy. And we can now see the structures in the light microscope and the nuclear in the confocal. And the development looks like this. So every 3 minutes, a picture was taken over, I guess, 3 or 4 days. And what you see, some microspores just die and do nothing, but some really go on proliferating and producing a lot of cells, as you have seen in the former pictures. And this process goes on until a larger callus is formed, and from this callus we can then regenerate plants. And of course, as scientists, we were interested in individual pathways. So you see here a whole variety of things which can happen in the individual pollen grains. But we were interested to follow that up individually and also to explain what maybe is the major pathway of these structures which can give rise to plants. And here is the example of one pollen grain which follows this pathway, which ends up in plant formation or has a potential of that. And this, in contrast to normal pollen development, is features a symmetric first cell division. If you remember, I told you the first pollen mitosis gives rise to 2 different cells, the generative cell and the vegetative cell. And according to these culture conditions, a big fraction of these pollen grains do not produce these 2 types of cells. They just divide symmetrically, giving rise to 2 same cell types. And of course, you need to look carefully. What you see here is one nucleus of the microspore, and this smaller green structure is the pore, the preformed pore of the pollen grain, where later the pollen tube comes out. And you should not confuse these 2. So here it is about the upper pollen grain and the larger nucleus. And when we look according to the development, you see the nucleus moves within the cell and all in a sudden divides in 2 nuclei, which have the same size, the same shape. And this proliferation now goes on simultaneously. They divide again. And so it goes on producing this multicellular structure. And when we counted the events within our culture, we came up with a little bit over 50% of all pollen grains following exactly this pathway. And altogether, we found 9 different pathways. At least 2 of them we assume can give rise to plants. But I will also show you another example, which also unfortunately happens quite often, that these cells do not survive the culture. And here, this pollen grain in the middle tries a lot to undergo cell division. And it takes a lot of time that something happens. But when something happens, you will see the whole cell messes up. And this we call programmed cell death. And you will see a burst of the nucleus forming lots of micronuclei, and the cell is no longer capable of dividing and surviving. And in doing so, we maybe coincidentally came across the observation of a very interesting phenomenon. And this is how the genome duplication can occur in this process. You may have heard about colchicine treatment so that you can obtain higher ploidies. But here we have the phenomenon that a fraction of the plants you produce are already have undergone whole genome duplication. In barley, that means they're diploid. And here we see the phenomenon which can give rise to this genome duplication when you look at the two nuclei with it. So here you see all nuclei in one pollen, which has an advanced stage of proliferation. And in the right corner below, you see a pair of nuclei, and you have to follow what they do. So, and together with light microscopic observations, we found that the cell wall between two newly formed cells is often not perfectly closed. And when these nuclei can get in touch with one another of these two newly formed cells, they fuse. And this is exactly what you see. And to show it one more time in a close-up, here you see these two guys again, how they get in touch and fuse to one diploid nucleus. And this is, of course, for us working in this field, a very exciting moment to see that marriage of plant nuclei. Yeah, and when we look closer, so these events can happen all over the time. So that can happen right after the first division or later. And then also chimeric plants can be produced when the later the nuclear fusion occurs. Of course, only a part or a sector of the plant is diploid, but you will see that because the diploid sector will give rise to seeds or grains, and the haploid part will not produce any. So now I come to another principle of forming haploids, and this is called uniparental genome elimination. Here you see also as representatives of parents only the egg cell and the sperm cell, and in certain constitutions of interspecific crosses or certain mutants which have the capacity to act as a haploidy inducer, these hybrids are produced. And then we see the phenomenon that the genome of one parent is eliminated again. And there are different mechanisms where maybe roughly saying the management of the two genomes does not correspond very well with each other, so that one genome accidentally gets lost. But of course, we have the benefit to produce haploid cells or haploid plants by that, because if only one genome is left, we get a haploid cell plant. And again, here we have to make sure that whole genome duplication is undergone, and then we will have double haploid plants. So, and those examples here are well known. So if you cross barley with its wild relative, Hordeum bulbosum, you will see this effect in a couple of events. When you pollinate wheat by maize, you will see this principle. And there are several other, maybe not so well-known examples. One of them is that it also works with potato when you use a certain wild species as a pollinator. But there's the other group of intraspecific crosses. So pollinators of the same species with a certain genetic constitution are also capable of serving as those haploidy inducers. And as such a system is heavily used within maize, it was also the first gene which was identified to be responsible for that. And this is a phospholipase, PLA1, given in red. And this PLA1 is mutated, and therefore this is the reason that the male genome is eliminated after fertilization. And also centromeric proteins can be responsible for this effect. The centromeric histone H3, when it's modified, this was found in Arabidopsis that it works. And in Arabidopsis, interestingly, you can use these mutants either as a paternal parent or also as a maternal parent, and it goes in both directions. So, and since this gene was identified, of course, we were looking whether there are orthologs in other species, and we found an ortholog of this PLA1 also in barley. And I will not stress you with the details given here, because I have many other things to tell. There's a second gene which also can contribute to that, but this is not of interest today. What we did, we mutated PLA1 by targeted mutagenesis using Cas endonuclease technology, as I presented yesterday, and we produced quite a number of those mutants, and 70% of the transgenics produced were mutated. And here you see where we selected target motifs within this gene, which comprises 4 exons. And below you see, again, the same principle. Upper line is a wild type, and below there are different mutations we obtained. And as you can see, smaller deletions, insertions of one nucleotide, larger deletions, quite a panel of different things we got. So, and we rendered those mutants homozygous and then pollinated. First we selfed by reproduction, and then we developed a method using these haploidy inducers to produce haploids in any barley or from any barley. So, and the method is that we have to emasculate and pollinate the maternal parent. And 7 days after manual pollination, it looks like this. These grains have already developed to some size. And since the endosperm is kind of handicapped, we need to rescue these embryos from collected caryopsis at that stage. But the embryo rescue works pretty well on a suitable medium. So, and when we selfed these plants, we already got some haploid plants. This was indicative of the method may already work. But of course, for research and breeding, it would be much more interesting to pollinate another accession with the haploidy inducers. And here in the diagram, you see the efficiency, what is the percentage, or what is the percentage of haploid plants amongst the plants you produce after such a pollination. And we started at the right side with Golden Promise, which is a selfing thing because we did it in Golden Promise. And this was about 6% of the plants were haploid. And then we used the 2 former reference genome accessions, Morix and Barker. And in Morix, we found a similar efficiency, and in Barker, surprisingly, it was much higher. It was 16%. And these are efficiencies a breeder can really work with. So, but we want to get one step further using this technology also for genome editing. And what I present here is a very specific approach where we transformed the haploidy inducer with a construct carrying Cas endonuclease and guide RNA. So in this case, we transformed maize with such a construct with a guide RNAs to be specific for a wheat gene. So they do not harm in maize at all. They do not find a target, and you can produce this maize, but you can pollinate wheat with this maize, and this is what we did. So you see here in the sperm cell, which is with this yellow interior, there's indicated the construct, the transgene construct inserted. And when we now pollinate wheat by this maize, this Cas endonuclease guide RNA all of a sudden has a target in the cell. And of course, the big question was, does it find it? And the concept was then that using this haploid technology, we know there's uniparental genome elimination. The parental genome will be eliminated after a short period of time where these 2 genomes were in one cell. So, and then of course, we would need to make sure there is whole genome duplication, and then we would have mutants which have no transgene anymore, or right away no transgene. So, and but this is not the major thing because these transgenes can be crossed out easily. But what for me was much more important is that that wheat transformation is pretty genome dependent. So we are not free in selecting wheat genotypes to be manipulated, and this haploid technology is much less genotype dependent, and therefore this was actually my driving force. Except that it's a cool approach. to look whether we can get it. And one other thing is, once you have such a haploidy inducer carrying this specific Cas-analogous and guide RNA, you can trigger specific mutations in a whole panel of different genotypes with this one plant even. So, and there was one concern when you imagine the different sizes of egg cell and sperm cell, egg cell coming from wheat, sperm cell coming from maize, that the sperm cell has not enough substance to bring in this endonuclease. And here I show you the dimensions of these two cells. So in my former scientific life, I prepared gametes of cereals. And what you see here is a pollen grain put in a mannitol solution, bursting because there is high osmotic stress, and it releases the two sperm cells, which is indicated above. And clever enough, we used transgenic material, GFP expressing material, so that we can see these two sperm in a confocal very clearly. And this told me that we can express Cas endonuclease efficiently also in sperm cells by using a ubiquitin promoter. So next question is the sperm cell big enough to bring in the active agent? And here I show you another part of my former life. When I isolated those gametes, I also did in vitro fertilization. And here you clearly see on the right-hand side, the egg cell is a huge cell and the sperm cell is actually a dwarf in comparison. And by the way, because it's also very exciting and it's also about haploid cells, I show you what we did here as a little side story. We were capable in an electric field to fuse these two cells with a little pulse of alternate current. And you see within a second, you see that these plasma membranes fuse in the middle, and in the right-hand side, you see the remnant of the sperm cell almost taken up by the egg cell, giving rise to a zygote. So, and the question whether the sperm cell is big enough or accumulates enough Cas enzyme was answered then by our experiments, where we then looked into the plants we generated, whether they have mutations. And in the beginning, it didn't work, but we worked hard, and in the end, we found some. And this is pretty busy, and I have to explain a little bit. It's always the same principle. The first lines are also always wild-type, but here are shown several groups of events. So we did it in common wheat, and we used two different genes, both related to the semi-dwarf trait I reported yesterday. So it is the brasinosteroid insensitive gene we mutated, and the semi-dwarf 1 gene, which is gibberellic acid oxidase gene, which also contributes to plant height. And as you can see, we did get mutations in common wheat and in durum wheat. And we, in all 5 genotypes we used, we got mutations, which is also a good sign that genotype dependence is not all too high. We got mutations in all subgenomes of wheat, A, B, and D. And we used 2 different genes and 3 different target motifs, and all gave us examples where it works. And this state of development was enough then to have a good publication about it, and to talk with colleagues where we could apply it. With that, I would like to conclude my talk, and thanks for your patience and attention.
Thank you, Jochen, for the wonderful talk. Ozgina o'zbekchada bularni anaqalarni aytib o'taman. Hozir o'sha bitta kichkinagina hujayradan, ya'ni chang ichidan o'sha o'simlik olishni gapirdilar. Bu juda ham ajoyib texnologiya. Yana bitta tarafi mikroskop bizada umuman yaxshi rivojlanmagan. Mikroskop bizada umuman anaqasi yo'q, texnologiyaga kirib kelmagan. Aynan mikroskop orqali biz juda ko'p narsalarni aniqlashimiz mumkin. Kimdirdir o'sha mikroskopik ishlar bilan qiziqishiga taklif qilganda, yo'q, bizada mikroskop umuman qiziqmas, hech kimni mikroskopik ish bilan ish qilishni umuman xohlashmagan. Lekin bu o'sha ko'rayotgan o'sha prezentatsiyada aynan mikroskopni kuchi juda ham kattaligini, bizga juda ko'p ma'lumot berishligini ko'ryapmiz, to'g'rimi? Keyingi anaqasida aynan eng qizig'i o'sha ikkita ikki xil turdagi o'simlik, masalan, bug'doy bilan makkajo'xorni chatishtirib, aynan oxirida bitta bug'doy o'simligini olish ham ajoyib texnologiya. Are there any questions? Savol?
Sorry, my English level. Hozircha tarjima qilasiz. Judayam zo'r prezentatsiya uchun tashakkur. Avvalo, Bubble Gaploid texnologiyasini paxtachilikda qo'llash bo'yicha savolim bor edi. O'zbekistondagi iqtisodiy ahamiyatga ega bo'lgan ikkinchi ekin g'o'za. Bugungi kunda yangi navlar yaratishga O'zbekiston paxtachiligi muhtoj, lekin bu uzoq 10-12 yilni talab qiladi traditsion metodika bo'yicha. Bubble Gaploidni g'o'zada qo'llashda nima muammolar bor va ularda yechimlar bormi? Hozir bug'doyda yoki makkajo'xorida qo'llagandek qo'llasa bo'ladimi? Yana mikrofon olmay, ikkinchi savol. Kas-9 edit bo'yicha target to'g'risida gapirishmadi. Off-target holatlarida yechimi topilganmi? 2024-yilda ham o'sha kerak emas joyda kesib yuborish holatlari yoki bunaqanday решение qilingan.
I did not understand a single word, but it was a pretty long question.
I will translate. So in our country, the second important crop is cotton. So it's possible to do play this method to cotton. First question. And if yes or not, in how?
Okay. I cannot promise. I didn't look into the literature where are already examples for cotton. That might well be, but maybe not. As I gave you the number, and the number is old. So I guess from the 90s, there were already 2,000 different plant species in which the regeneration from isolated microspores or anthers containing those microspores give rise to haploid or double haploid plants. So the chances are pretty good. And I can also give you maybe another hope. We are currently working in several species which have not been that important in history, but now become more important, like legumes or pseudocereals. And for all those, there is no such technology available. But having three choices, using microspores or parthenogenesis, ovioculture, ovariculture, or finding another related species, when you pollinate, you may get haploids, you have lots of chances. And this is what we do. So we don't know whether we are successful, but we know the principle is pretty universal. And therefore, it's always good to give it a chance, work hard, and maybe you will be the successful one.
Men aynan bu texnologiya bilan ishlagan olim sifatida ayta olishim mumkin. Bu narsa mumkin har bir o'simlikda, faqat savol biz o'sha to'g'ri optimal bo'lgan condition sharoitni yaratib bersakgina u o'sha bittagina kichkina tiny single hujayradan bitta o'simlik olish mumkin. Buni iloji bor. I will he has a second question was
maybe one comment.
Yes.
Since I know that Uzbek people are pretty smart and hard workers, I have a Uzbek guy working exactly on this in the pseudocereal quinoa, which has been an orphan crop, but now gets a lot of importance. And if we want to unlock this species for modern breeding, we need haploid technology. And Mohammadion is working exactly on that in quinoa.
His second question was hozir kunda o'sha eng qiyin, endi nima desam, muhim bu ahamiyat kasb etayotgan o'simliklar bor. Oldin umuman e'tiborda bo'lmagan, hozir kunda juda e'tibor talab qilyapti, chunki ularda oqsillar ko'pligi yoki qaysidir ma'noda oziq-ovqatda o'sha nima desam, bir o'zida kerakli anaqa vitaminlarni tutgan o'simlik bo'lgancha, hozir kunda unda o'sha gen injeneriyasi yoki boshqa texnologiyalar qo'llayapti. Bizada o'sha Muhammadjon Mirzahmedov, ya'ni Center genomikada ishlagan. Hozirgi kunda kino o'simligi ustida aynan double gaploid texnologiyasini yo'lga qo'yish va transformatsiyasini yo'lga qo'yish bo'yicha ishlayapti.
Quinoa.
Yeah, no. The second question was but it's the question actually not related to directly double coupler technology. This was related to genome editing. Like, how about off-target analysis? Is there any kind of solution? Actually, how we can cope up with this off-target effect?
Off-target mutations are mainly seen as a problem. I do not see that as a problem. Maybe a safety problem, not. Maybe for breeding, it can be a problem if you have an unexpected effect, which is deleterious. Of course, we need to care on that. But off-target mutations produce additional genetic diversity. And we have experienced in cooperation with Sophia, a very nice example where we mutated the nudum genes to produce naked barley, as reported yesterday, and we have off-target mutations. In the beginning, we did not really care on that because I thought, okay, there are mutations all of the time. So why we should care on these specific off-target mutations? But by then carefully looking at that, we found another family member of this gene mutated. And this initiated us to have an entirely new story on Vex formation at the plant surface. And this is for me a very good example. So there is no safety issue because there is always spontaneous mutagenesis in all organisms. And of course, this has a certain risk, but when you have off targets, there is no reason to regard these as more dangerous as any random mutation. And random mutations appear to happen much more often than off targets appear to happen. And therefore, the risk is not even comparable. It's like a drop in the ocean, you know? And I see it much more as a chance that we create additional biodiversity, which we can use in science or even in breeding. But of course, I have changed my mind. We should not disregard these off-target mutations. We should look at them, maybe to profit the best way out of them.
Off-target analiz bo'yicha asosan aynan yaqinda o'zimni o'sha maqolamda qilganman. Siz o'sha guide RNKlarni tanlayotganingizda genni siz o'sha tanlangan guide RNKni tekshirib ko'rish kerak. Juda boshqa genni o'chirib qo'ymaslik, ya'ni boshqa joyga to'g'ri tushib qolmasligini oldin tekshirish kerak. O'shanda agar sizda hech qanaqa genga o'tirmayaptimi, demak sizda hech qanaqa off target mutation bo'lmaydi. Lekin men nima qilganman, birinchi уже transformatsiya qilib, уже menda mutatsiya bo'lgandan keyin men off target analiz qilganman. O'shanda men ko'ryapman, men boshqa genni ham o'chirib qo'yganman. Qaysidir ma'noda to'liq emas, qanaqadir 70% bu yerda ya'ni functional bo'lgan tarafi ham bor. Agar domla aytayaptilar-ku, siz o'sha bilmagan holda boshqa genni o'chirib qo'yib, qaysidir ma'noda foyda olishingiz ham mumkin. Lekin buni baribir ko'zdan qochirmaslik kerak. So thanks so much, Jochen, for the interesting discussion. Now I think, sorry, Jochen, sorry.
Assalomu alaykum. Hurmatli anjuman qatnashchilar. Barnopa boyaga so'zimizga to'ldirib ketaman. Bizda mikroskopiya tadqiqotlar yaxshi rivojlangan. G'o'zada asosan rivojlangan, bug'doyda u qadar emas. Ya'ni genetik tadqiqotlar demak dunyoda ikkinchi o'rinda turadi. Bizda demak tadqiqotlar Milliy universitetda genetik kolleksiya bor g'o'zani. Sanamyan va uning shogirdlari tomonidan yaratilgan. Hozirgi ushbu genetik va molekulyar genetik tadqiqotlarni birlashtirgan holatda biz ham gaploid texnologiyasini yaratganmiz va oldik va Xilzotum turida birinchi marotaba dunyoda gaploid oldik. 5 ta gaploid o'simligimiz bor, har xil demak boshlang'ich materiallar asosida va 2 ta demak double gaploid liniya hozir olganmiz. Bundan tashqari yana o'sha genetikani yutug'i bizning respublikamizda, xususan biz laboratoriyamizda biz o'sha genetik kolleksiyadan unumli foydalangan holatda demak yangi xromosomasi almashgan liniyalar yaratganmiz. Bu AQSh kolleksiyasida mavjud emas. Shuning uchun menga taklifim kelgusida aynan qaysi o'simlik ekanligini fiksatsiya qilib o'tsangiz yaxshi bo'ladi. Bug'doyda u qadar rivojlanmagan. mikroskopik tadqiqot, lekin g'o'zada yaxshigina rivojlangan. Mani savolim bordi bir necha. O'zbekcha beraveram. Birinchisi ruscha tushunmaydi. Birinchisi, qaysi metodlar qulay? Ya'ni gaplotexnik olish uchun. Birinchi bu spontan, tabiiy ravishda, ikkinchisi sun'iy ravishda turlararo, turkumlararo chashtirib gaploidlar olish va uchinchisi aytganda changchilar yordamida gaploid o'simliklar olish. Lekin bu metod hamma o'simlik uchun qulay emas. Xususan g'o'za uchun, ya'ni changchilar orqali olimlar tomonidan bir necha tadqiqotlar amalga oshirilgan, lekin muvaffaqiyatli chiqmagan. Changchi yordamida gaploid olish. Birinchisi.
To'liq anaqa
Uchta metodda qaysi biri qulayligini aytib bersanglar bo'lsa.
Jochen, what you described in your on your talk, so there are like several methods to get to obtain the double gaploid, right? And in cotton, which method might be or can be suitable?
This is not predictable, but my advice is to start with this pollen, because you have so many pollen in most of the plants that the chance is good that at least a few of them decide to follow this pathway in contrast to normal pollen development. And in history, actually, the majority of all viable methods are based on this principle. And only if you fail with it, you have these alternative things with parthenogenesis and genome elimination. First is always microspore culture, answer culture, following the principle of pollen development.
Thank you. İkinci savolim как раз elimination of chromosomes. Bu yerda rus olimasi Pershini 3 stadiyaga bo'lgan eliminatsiya xromosomasining genom, elimination genom, ikkinchisi ya'ni частичный eliminatsiya, ya'ni bir necha xromosomlar yo'qolib qolishi, uchinchisi alohida xromosomlar eliminatsiyaga uchrashi. Bunga nima sababli bo'ladi? Ya'ni H3 oqsildan tashqari.
The question is the elimination of elimination of chromosomes, and then how does elimination happen? Sometimes the one chromosome eliminated, or at the same time, or multiple chromosomes eliminated, and what could be the reason for the elimination and all different types of elimination?
Okay. Yeah, this is pretty complicated, and therefore I did not want to bother you with that, but I try to make it short. So there are different mechanisms that one parent's chromosomes are eliminated. And as I tried to generalize is that the chromosomes of both parents are processed in one cell simultaneously. And as these chromosomes come from different species, the processing and reshaping and undergoing cell cycle and so on is not simultaneous when you have a hybrid. And this is also a reason why in many crosses no hybrids can be obtained, because one genome is eliminated and then the genome of one parent is left alone and you have no hybrid. So one more particular mechanism is when a chromosome undergoes development, it forms an active centromere, which can be taken by the mitotic spindle to be drawn away as a chromatid from the other to be placed to one daughter cell and to the other. And the centromere is not formed or still not mature enough in one parent's chromosomes. And so these chromosomes are not allocated to a daughter cell. And this is a mechanism which is proven. But we guess there are also other things playing a role, but this is just an example how it can happen.
Thank you. Oxirgi savolim, ya'ni bu ham gaplar o'tuvchi xitoyliklar CRISPR-Cas texnologiyasidan foydalangan holatda DMP genlarini o'chirganda va gaploid o'simlik oldi. Ya'ni g'o'zani gasepxizotini turdi. Ammo bu genlarni o'rgansam, bu genlar oilasi g'o'zada qurg'oqchilikka ham javob berar ekan. Hozirgi kunda g'o'zada qurg'oqchilik bu ayni aktual muammo. Gaploid texnologiyasidan olish Ya'ni buni asosiy maqsadi biz double gaploid olamiz va seleksiyaga qo'llaymiz. Seleksiyada qo'llagan quruqchilik chidamsiz qolsa u qanday nav yaratsa bo'ladi? GMP genini kirib qo'ydi. GMP prezentatsiyada ham ko'rsatdilar.
There are gene which called DMP. It is responsible to induce the double couplet. And if he said when he searched the gene, it belongs to drought related.
It's a whole gene family.
Yeah.
It's difficult to spot the most related ortholog. Yeah, I agree.
Like switch the function of the gene, and we have a double couplet plant, but we lost the drought tolerance. And what is the kind of advantages of doing this? So because drought is also really kind of main problem in our country because of the dry condition. So
I don't understand the question. When you have a haploidy inducer, and this haploidy inducer is mutated in PLA1 or DMP, then this genome is not included in the plant you produce. So this is exactly the mechanism that the genome of the haploidy inducer is eliminated. So all these genetics do not play a role in the double haploids produced. So you can mutate whatever you want in this inducer line because it doesn't contribute to the genetic makeup of the plants you produce.
Tushunarli. Thank you.
yeah, thanks so
Thank
much.
you for your interest.
Yeah. Now time to break. So we have a coffee break now. Then we will continue after the coffee break.
Bir, iki, üç.
Yeah, welcome back. Now I hope you have you gained energy during the coffee break. And now Wendy is continuing his second talk following the first presentation.
Thank you. So in my previous presentation, I spoke mostly about genetic discovery, using genetics and genomics and bioinformatics to discover genes. And this is what I've been doing for the last 15 years or so in disease resistance of cereals. About 5 years ago, I moved from the UK, where I had my lab in the John Innes Centre. I moved to KAUST. And with the generous funding that I now have at my disposal in KAUST, I set up 2 additional pillars of research in my lab, which are depicted here. So we have our discovery and function. We still continue that. And then we're also venturing out into mechanism. This is a very new area for me. And we're also now starting to do some engineering. And then finally, we have a pathway to impact partner for our deregulation and deployment. And so in this second presentation, I will talk about some of the work that we've been doing in KAUST over the last few years in the area of mechanism. And so by way of background, I mentioned in my previous presentation that most of the genes for disease resistance that have been genetically defined and subsequently cloned, they belong to 2 classes of resistance gene structures. They typically encode surface receptors consisting of leucine-rich repeats that act like aerials on the surface of the cell, monitoring the environment for the presence of non-self. These leucine-rich repeats are protein-protein interaction domains and can detect the presence of pathogens based on effectors that they secrete. And then they have a transmembrane domain, and some of them may be decorated with a signaling domain, a kinase. And then you have a second type of receptor, which is an intracellular immune receptor, a so-called nucleotide binding site leucine-rich repeat domain. And if we look at the number of genes that have been cloned over the last 30 years since the first one was cloned, you can see this is a sort of slightly exponential growth. And to date, more than 400 disease resistance genes have been cloned. And we looked at all of these genes and quantified the structures that they encode. And as you can see, most of them are cell surface receptors or these intracellular NLR receptors. And then you have some oddballs, some other things. And we've seen that there's a class which is emerging, which are these kinase fusion proteins. And if we look at where these disease resistance genes have been cloned from, it's interesting to note that while many genes have been cloned in the model species Arabidopsis, the cereals are now leading the way. And these are primarily genes that have been cloned in wheat and corn or maize. And I find that very gratifying now that we can do a lot of this work that was only possible before in Arabidopsis. We can now branch out into the major crops and clone these genes. And here's another analysis that we submitted when we wrote this review. We submitted it to the journal, but one of the reviewers did not like it. It set off a lot of antibodies. And this is where we quantified the impact of, the publication impact of cloning these genes. And so on the y-axis, you have the citations normalized by the number of years. And I'm just trying to say that there's still a lot of interest in cloning these genes, and you can be creative in the way that you publish them and still publish in good journals. Okay, so by way of a little further introduction, and this relates, I think, to the question that Staffan asked at the end of my first talk, and that is, how do these receptors work? And the most simple model is that they detect the effector from the pathogen. So the pathogens, they secrete a lot of proteinaceous molecules, either into the apoplast of the leaf, or they have ways of injecting these proteins into the plant cell, and they promote pathogenesis. They subvert the cell and help the pathogen to grow and reproduce. And of course, the plant has evolved these immune receptors that can detect the presence of the pathogen and elicit a defense response. And sometimes this is a direct interaction where the immune receptor will recognize the effector and elicit a defense response. But a very important contribution to understanding what's going on was made by Eric van der Biesen, a postdoc in Jonathan Jones's lab in 1998, and he proposed the so-called GARD hypothesis. And in this model, the effector is not recognized directly. The effector is interacting with a pathogenicity target, which is shown in orange, and the immune receptor is guarding this pathogenicity target and can detect modification on the pathogenicity target imposed by the effector. And this can then turn on downstream defense responses. And this is a very nice model because it explains how millions of effectors from lots of different pathogens can be guarded by a limited number of immune receptors. A typical plant genome will have from a few hundred to a few thousand immune receptors. And if you now picture in your mind that these immune receptors are guarding cellular hubs, there's only a finite number of hubs that the host would need to guard. And a corollary of the GARD hypothesis is the so-called decoy model. And in this model, the pathogenicity target has been duplicated in the genome, and that releases it from the functional constraints that it may have. And so now the immune receptor is recognizing a decoy, and the decoy is acting as a trap for the effector. And finally, in the integrated decoy, the decoy is physically integrated into the gene of the immune receptor. So you have the decoy and the immune receptor right next to each other, physically next to each other. And that occurs in about 10% of NLR. So 10% of NLR immune receptors, they have integrated decoys. And this tells us a lot about the types of pathogenicity targets that effectors they target. Okay, so we started off years ago cloning disease resistance genes in wheat. And I will admit that at the time I was expecting that we would clone NLRs, these intracellular immune receptors, which are the most abundant genes that have been defined by genetics and cloned across many different systems, including in monocots and dicots. And to our great surprise, many of the genes that we cloned in cereals, they did not fall into this class. Instead, they were kinases. Very, very often they were tandem kinases, so two kinases fused together. And sometimes they had an altogether completely different exotic domain fused to them. And we call these class of genes now, we call them kinase fusion proteins. And of course, we were wondering, how do they work? Right? What's the mechanism? What's the modus operandi? And so that is what I will talk about in this presentation. We have focused our attention on this gene here, SR62, and I'll take you through how we studied SR62 and determined its mechanism. So SR62 is a gene that was introduced into wheat from a wild grass, Aegilops sharonensis. It confers very broad spectrum resistance to stem rust, so it's a very good gene. And it was cloned by a postdoc in my lab, Gu Taiyu, and we published that in 2022. And as part of the, this looks a little bit corrupt, this slide. Anyway, as part of the gene cloning procedure, we generated EMS mutants. And you can see the effect of these EMS mutants on the mature protein up there. And SR62 encodes a wheat tandem kinase. But as part of cloning SR62 and generating these mutants, we also found a few mutants. We found 4 mutants that did not have a mutation in SR62. And these are depicted here, 119D, 200H, 267D, and 1298E. The other 2 are controls. We've got the resistant parent, so here's 1644, which carries the intact gene and on the far left. As you can see, that's completely immune. And then we have the recurrent parent, so here is completely susceptible. And the mutants, they are as susceptible as Sahir. And so the question was, if they don't have a mutation in SR62 in the kinase, what are they mutated in? And my former PhD supervisor, Jonathan Jones, he has followed somehow in my footsteps. He has become an adjunct professor in KAUST, and he comes out to visit KAUST every 2 weeks. Every year he comes for 2 weeks, typically in December. It's very nice. We catch up. We talk a lot of science. And I was telling him about this project. We were sitting outside under the palms, having a lovely lunch. And I told him about this. I told him about these extragenic suppressors. And he said, if it's an NLR, it's a Nature paper. So, of course, I was very motivated then to clone this gene. And so postdoc and 2 students in my lab, Renji, Karthik, and Oliver, they set about cloning this gene. And to cut a long story short, we found that it was indeed an NLR. So the tandem kinase and this NLR, they occur physically close together in the genome as a digenic module. So we have the kinase and the NLR very close to each other. And these 4 mutants, we could indeed show that they were all in this NLR, providing very good evidence that this is the gene that is required for the function of the NLR. Okay, in parallel with this, we worked with Peter Dodds and 2 postdocs of his, Megan and Sean, in the CSIO in Australia. And at the time, they had developed a very good system for cloning the corresponding effectors of resistance genes. And they applied their method to SR62. And lo and behold, they identified the corresponding effector. So they identified AVR SR62. And AVR SR62 belongs to a multi-gene family in the stem rust fungal genome. And you can see in this strain of the fungus, on chromosome 5A, we have 4 copies, and on chromosome 5B, we have 3 copies. And I've highlighted the ones in green that are able to elicit the hypersensitive response, that are functional versions of the effector. And they were able to validate these in a protoplast assay. So they could isolate protoplasts from wheat and deliver the corresponding effector with the wheat tandem kinase and elicit a hypersensitive cell death, which is measured by quenching of a reporter gene that gives fluorescence in this system. And also, interestingly, they showed that the tandem kinase is the primary immune receptor. They used yeast-2 hybrid to show an interaction, a physical interaction in vivo between the tandem kinase and the immune receptor, and the effector. Notice that only the green flavors of the effectors, the ones that are able to elicit a cell death response, only those physically interact in the yeast-2 hybrid system. Okay, the next step was to functionally validate the NLR and its requirement in this system. And we used the protoplast system, and we were able to take mutant backgrounds that had a mutation in the NLR and deliver to these mutant backgrounds the NLR and the effector. And only when we delivered the functional NLR and a functional effector were we able to elicit a cell death response and quench the reporter gene activity. So this was very nice, a very quick way to validate the function of the NLR. And we were also able to recapitulate the gene-for-gene interaction in a heterologous system by transiently expressing these components in a leaf of Nicotiana benthamiana. And so here, in each of these sectors that are indicated by the circles, we have expressed the NLR and the tandem kinase. And if we express these 2 together, with a negative control, YFP, which you can see at about 1 o'clock in the top right-hand corner, you see that there's no cell death. So just expressing the NLR and the tandem kinase, you don't get cell death. But when you co-express the green flavors of the effectors, you get cell death, but only the functional effectors. Indicating that we can recapitulate the gene-for-gene interaction in this transient system, which is very useful because this is a very fast assay. Okay, so now I'm going to take you through 3 slides, which will give you an idea of how we dissected the mechanism. Just going to show you 3 slides, okay? So keep your attention for 3 slides. I'm not going to take you through the 70 pages of supplementary data that was in the paper. Okay, just 3 slides. So in this first slide, and this was an experiment done by Luby and Renji, 2 postdocs in the lab, they showed that in the resting state, the tandem kinase and the NLI immune receptor, they do not interact. So if we fuse the partners with a reporter gene, luciferase, we do not get illuminescence when we express these, which is shown on the left-hand side of this leaf. Okay, so the kinase with the NLR does not interact in the resting state. But when we co-express a functional AVR effector, AVR SR62, then the NLR is recruited to the complex, and you get functional luciferase expression and light. Okay, so this is very nice. And again, it was a very fast assay to do. And when we then did yeast2 hybrid and separated kinase 1 from kinase 2 in the wheat tandem kinase, we could show that it is kinase 1 which interacts with the effector. It's not kinase 2. Kinase 2 does not interact with the effector. Okay, so then we went on, and this is work done by Sean in Peter Dodd's group, and he showed that AVR SR62 is able to disrupt the interaction between kinase 1 and kinase 2. So first of all, he in this assay, it's a plant 2 hybrid assay in which you get the accumulation of a red product called betalin when 2 proteins are physically interacting. And when he expresses kinase 1 and kinase 2 separately in trans, he gets interaction, which is shown in the top right-hand corner. You get the production of this red pigment showing interaction. However, if you co-deliver a functional effector, then kinase 1 and kinase 2 dissociate from each other, and you break that interaction. Okay, so to recapitulate, in the resting state, kinase 1 and kinase 2 are interacting, and that interaction is lost when you add a functional effector. But the interaction is not lost when you add a non-functional effector, as shown at 7 o'clock in the leaf. Okay. The other thing which is very interesting is that you can express kinase 1 and kinase 2 in trans along with the NLR immune receptor. And when you express kinase 2 with the NLR, which is shown at 11 o'clock, you get an HR. Okay, so kinase 2 is enough to activate the NLR and give rise to a hypersensitive response. However, if you co-express kinase 1 and kinase 2 in trans together with the NLR, that HR, it doesn't happen. So in other words, kinase 1 is negatively regulating kinase 2. And you can de-repress that regulation by delivering in trans a functional version of the effector, which is shown at 7 o'clock. When you deliver a non-functional version of the effector, that de-repression does not take place, as shown at 4 o'clock. Okay, so I said I would cut the mechanism short, and I'm going to go directly to the model that we came up with. And there were a myriad more experiments that we did, and the reviewers doubled the amount of experiments that we did. So this model is very well tested. And of course, you can go to the paper and you can read all the detail if you wish. But to cut this long story short, we found that in the resting state, the wheat tandem kinase exists as a homodimer. And in this homodimer, kinase 1 is interacting with itself. So it's interacting with kinase 1. And kinase 2 is interacting with kinase 2. And also kinase 1 and kinase 2 interact with each other. Okay, so they're tightly bound together. like this. And then when you add the effector, it comes along and it interacts with kinase 1, and this releases kinase 2 from this complex. It's still tethered, but it's now floating about. And this exposes an N-terminal lobe in kinase 2 that allows it to recruit the NLR to the complex. And this recruitment happens through an N-bark domain, which is highlighted in red in the figure. And this then activates the NLR and leads to immune responses. And we coordinated our work with a group in China, in Beijing, and they cloned the same NLR as us. And so we were a little sort of worried about the competition, but actually it was a really, really good coordination, and we managed to publish back to back. And what was really nice was that they worked on the latter immune responses, and they showed that, so they worked on all the late molecular interactions, and we worked on all the very early molecular interactions. So it was very nice to have these two papers back to back in the same issue. Notice that this kinase 1, it has these long bits, and these are called extended beta fingers. So these are it's an extension of the beta finger in the kinase domain, and this extended beta finger is responsible for maintaining the homodimer as a complex, and it's also the bit that interacts with the effector. And so here's a result from our study that I'm very proud of. I encouraged Oliver, who's a bioinformatician, to download serial genomes, and he downloaded 70 serial genomes, and he managed to find 30 or so orthologs of the SR62 kinase. And he aligned all these orthologs with each other and was able to identify regions of the protein that are subject to natural variation. And when you project that natural variation onto the AlphaFold generated 3D structure of the wheat tandem kinase, which is shown here as a homodimer, what is really interesting to see is that the hotspot of variation occurs where the effector is binding. So it would appear that there's been selection for variation at the recognition surface. So there's really an arms race going on between the effector and the immune receptor. And if we draw the extended beta finger as a ribbon instead of a blob, and that's shown on the far right, you can see the beta 1 strand going out and the beta 2 strand coming back. And you can see that the beta 1 strand, which is the one that's buried in the complex, is rather conserved. And this is the one that is mediating the interaction between the kinases. Whereas the beta 2 strand, which is on top and which is solvent exposed and is the one that's interacting with the effector, that is the one that is really hypervariable. Okay, so then this is what Jonathan, my former PhD supervisor, remember this is what he said when he came to visit in December 2023. Then he came back to visit again in December 2024. And I told him, Jonathan, we've cloned the gene. It's an NLR. And then he said, it's a science paper. So, you know, he's getting old a bit. He's now 70. He's just turned 71. So maybe this was a senior moment. But anyway, we followed his advice and we submitted it to Science. And I was very happy. It was a positive outcome. Okay, so you may ask, you know, why all this mechanism? What's the relevance for breeding, in particular GM breeding? And so to remind you, SR62 was integrated into wheat from Aegilops sharonensis. And if you have the two Sharonensis versions of the SR62 kinase and the NLR, they work very well together and they give rise to immunity. But if you were to put SR62 into a stack and express that in the cultivar fielder, it does work. So the fielder NLR is able to work with the Sharonensis version of the kinase, and you can get immunity. But we have shown that if we take another flavor of the NLR from another cultivar of wheat called Sahir, these two do not work well together and do not give rise to immunity. So it's very important to have the two right combinations of this digenic module together. And so what we've learned from this exercise is that if we're going to build a stack, it's probably a good idea to include the kinase and the NLR together in the stack. Okay, so continuing a little bit further with this interesting feature that we discovered, the extended beta finger, we looked at other kinases that have been cloned as major determinants of disease resistance gene in cereals in the last 20 years or so. And it's not just us who have cloned some of these genes. The community as a whole, in the last 5 years, there's been a lot of these genes that have been cloned. And we looked and we found that in nearly all of them, except one, there's this extended beta finger in the kinase. So we thought it must be an important feature. And so Oliver, he built an HMM model, which is able to recognize these features in a plant genome and annotate the plant genome. And as you can see, there's quite good conservation in the first few amino acids and in the last few amino acids, and a little bit of conservation in the middle. But apart from that, it's a very variable feature, this extended kinase. The length is very conserved, but many of the positions are very variable indeed. And within the wheat genome, we've annotated all of these now in Chinese Spring, and they are scattered throughout the genome, mostly found in the telomeres, where in the gene-rich region. And many of them occur quite close to an NLR. So it gives us a possible hint at which NLR these kinases might be working with. And this is something we can now, of course, empirically test. And this shows the type, the phylogeny on the right shows the type of kinase fusion proteins with the known ones shown on top. Okay. Now, what's interesting is that some of these fusions appear to have evolved independently. So here we have shown, we've highlighted in green the kinases that are fused to motile sperm proteins, MSPs. And you can see two very distinct clades. This phylogeny is based on the extended beta finger. And you can see two very distinct clades, suggesting that possibly MSPs were recruited independently at least 2 times during the evolution of wheat. And here's another example of two kinases that are fused together. And this fusion also possibly occurred multiple times. And also interestingly, many of these extended kinases, they occur in kinases that are singletons, that are not fused to another kinase or to an altogether exotic domain. And they may be the progenitors that through evolution recruited these exotic domains, which we believe probably act as decoys, following the decoy model that I presented early on. So they're a bit like integrated decoys, but not for NLRs, but for kinases. And another very important feature that we've discovered is that if you look at the expression of these kinase fusion proteins in a plant genome, the ones that have been defined as being functional are invariably in the top 25% most expressed kinase fusion proteins in the transcriptome if you just look at kinase fusion proteins. Okay, so this is a hallmark, a feature that could be used to speed up the cloning and identification of kinase fusion proteins. Because if you have an interval that you have genetically defined and you then project that genetic interval onto a physical genome, you can look at, annotate the genes in that region and look at their expression to help identify the best candidates. And if we look at the genomes of monocots, we find that this feature, the extended beta finger, is restricted to the Poaceae. So suggesting that the feature evolved about 100 million years ago. And this may explain also why disease resistance genes encoding these kinase fusion proteins have only been identified in wheat and barley and their wild relatives so far. And this work hasn't been published yet in a peer-reviewed journal, but you can find this story explained in this bioRxiv paper. And so a few concluding remarks. The SR62 kinase and NLR, they function as a digenic module encoding a sensor and a helper, where the sensor is the kinase fusion protein and the helper is the NLR. And kinase fusion proteins, including SR62, they have these extended beta fingers, which are near ubiquitous. We've only found one example of a functional kinase fusion protein that doesn't have this extended beta finger. And this feature is private to the Poaceae, and so most likely evolved about 100 million years ago. And functional kinase fusion proteins have so far only been cloned from the Triticeae. So that's the group of grasses that include wheat, barley, and rye and triticale. And once again, this is the acknowledgements to my group who conducted this work. And I'd also like to thank again Peter Dodds and his group in the CSIRO in Australia, and the very good collaboration and coordination that we had with our friends in Beijing, Xiyong Lu and his research scientist Ping Lu. And thank you for your attention.
Yeah, thank you, Brandy, for the this is great achievement and exciting research. And I think we will have a lot of questions, I hope. So who will be starting? Is there any questions?
Thank you. While folks are thinking over the amazing talk you just gave, I thought I might start out with a couple of questions. So is the kinase not a GARDI, or can it be a GARDI? Thought of as a GARDI, based on your earlier talk?
Yes. So what sort of model does it fall into? It's a good question. I would say, so we're treating it more as an integrated decoy. So we suppose that the effector is targeting something else in the genome. It might be another kinase, and this SR62 kinase has become a trap, and is able to trap the effector into triggering an immune response. But this is not something we have proven. This is a hypothesis, and I think we would have to understand what the effector is interacting with in the proteome, in addition to the kinase fusion protein.
Right. So I wanted to ask you, following that, like, is the kinase activity important for how it works, or?
That's a very good question, and it was something the reviewers asked for also. and we blushed over that and said that this was beyond the scope of the paper. But we are now studying that, and we have found that kinase 1 has strong kinase activity in vitro. And kinase 2, on the other hand, does not have kinase activity in our assay, and this is what you would expect because it's missing some of the important residues in the kinase. We're beginning to unravel the role of this kinase activity. We found that it phosphorylates a particular residue in kinase 2. And if we knock out, if we replace that residue with an amino acid, an alanine, I think it is, then we abolish function. We don't yet understand how this is important for activating the NLR.
Thank you.
Yeah, thank you again for your second nice talk. So decoy, if I have understood correctly, must acquire specificity to a certain effector. And of course, you did not go so much into detail, but probably you, or very likely you have looked at the kind of generic structure, how this specificity can be attained, like leucine-rich repeats or other kind of antibody principles where modules are shuffled to create a whole variety of possible specificity. And
is this the great variability that we see in the extended beta finger? Is that under diversifying selection to create a scaffold? Is it a shapeshifter that can interact with effectors? That is a direction of travel that we are exploring.
Go ahead.
You put your finger right on it, Jochen.
Yana savollar bormi? Agar savollar bo'lmasa, bu yerda bir narsani aytib o'tmoqchi edim. Agar prezentatsiyada e'tibor bergan bo'lsanglar, juda ham ko'p o'sha Nature maqolalarni ko'rsatdi. Bularda o'rganadigan narsamiz ularni o'sha qilgan ishiga biz hali yetishimizga ancha bor. Lekin bularda yana boshqa narsa bor. Shunchalik zo'r ishlar qilib qo'ysa ham bulardagi oddiylik, bizadagi o'sha qanaqadir odamgarligi, o'sha siz qanaqa yordam so'rasangiz bular jon dildan sizga yordam berishligi, oddiyligi, odamiyligini saqlab qolgan holatda o'sha bu narsa juda ajoyib, biz o'rganadigan narsa deb o'ylayman. Chunki bizada agar shu darajada maqola qilsa, bizada o'zidan katta qanaqadir ma'lumot nimadir yasab oladi. Men hozir buni ingliz tilida aytmayotganim, olimlar ichida Nature'da chiqarmaganlari bor. Hozir maddohlik bo'lib ketmasin deb bu narsani faqat o'zbek tilida o'zim gapirdim. Thank you.
Thank you.
I'm currently inviting our next speaker, Kanat Yermekbek-White. And actually, I met him 8 years ago at the conference of Plangin in Kazakhstan. At the time, he was going to London to do his PhD, right? It was
Norwich.
Yeah. And then now he's already leading his own group. And he's head of the molecular genetics and genomics. And he will be presenting something single set to Kittrell and beyond and Triticum. Looking forward to your talk.
Thanks, Vanna, for the nice introduction. So, of course, it's very difficult talking after the person who only published in Nature and then now started publishing in Science, but I'll try my best. So we are quite excited what we are doing with, and we think that we will add some values to the real breeding practices locally in Kazakhstan. and beyond the country. So title just states from single seed to QTL and beyond. First of all, I'd like to thank organizers for the nice invitation and being considered toward me. As I said, Barna said, I'm Kanat, and now I'm leading a lab which is called Genetics and Genomics at the Jetsu University, which is located in Talkhorvan city, not far from Almaty, which is the biggest and the previous capital in Kazakhstan. And the university is called Jetsu University, and there is a small institute called Institute of Biotechnology and Ecology that consisted of 4 laboratories. And the reason why I went this small university, they are quite ambitious, and they promised a lot of funding, of course, and we are not as rich as King Abdullah University, but they provided enough funding to set up my lab, and eventually they built a new building for the institute. So this is the background of this talk. The main motivation of this talk is my PhD research, which I did at John Innes Center under the supervision of Simon Griffiths, who received a prestigious award this year for his excellent work on Watkins collection. I'll be talking about a little bit about that Watkins collection in my second talk. And the main, this is the main outcome of my PhD research. So the main outcome is we identified and mapped the 2 height controlling QTL in Central Asian wheat. So this is just a background information, as we have some junior scientists here. So the plant breeding is a kind of art or science, selectively mating 2 contrasting genotypes that has a contrasting phenotypes in order to create a mapping or kind of segregating population. And why we do? Because this is global food security. We need more food. Of course, then we also want that food to be long-lasting and looking beautiful. And also we need our crops to be resistant to abiotic and biotic stresses. So simple scheme, selection of parents and crossing, generating hybrids, testing and releasing new variety. This takes up to 14 and sometimes 18 years, which we call kind of traditional breeding. And within that traditional breeding, you just select only for goods, just eliminating the bads. And at the end, you end up with the good, adaptable, and high-yielding genotype. However, in our current understanding, good variety is a combination of good alleles. At most important genes such as VRN, PPD, RHT, or any other genes that confirm the genotype to be resistant to abiotic and biotic stresses. In order to have this nice combination of good alleles, only traditional breeding is not enough. Unfortunately, in our region, I think including the Central Asia, all the developing countries, we cannot do some kind of sophisticated genetic analyses, and all the breeding processes based on traditional breeding. So this slide shows a roadmap to do a kind of modern plant breeding. In order to identify those nice alleles, good alleles, we need to generate a kind of rich diversity, and that there are several ways. As Brenda said, there is a EMS method to generate the diversity, or the diversity panel, which we inherited from our ancestors, the old cultivars and the land races, or any other interesting genotypes. Also, we can create kind of multi-parent advanced generation intercross, which we call magic populations or nested association mapping populations that comes from the several inbred recombinant inbred lines. When you have this rich genetic diversity, you will have that the phenotypic and the genetic data. At the end, you just integrate those great deal of genetic and phenotypic data and obtain a signal in the DNA that is associated with the adaptation and its components. Despite Kazakhstan being a major, not giant, but major wheat provider globally, specifically regionally in Central Asia, as I said before, all our breeding processes are based on mainly traditional breeding schemes. This is also worrying if we consider the geopolitical shifts taking place in the world, like conflict between Russia and Ukraine. It poses a great threat on our global food security. So that's why I read one paper, but I couldn't find that one. So we have to take advantage of developing countries which has a agricultural capacity like Kazakhstan in order to secure our global food security. Because of the methodologies we are using, as you can see from the graph, an average wheat yield in Kazakhstan just stagnated since the beginning of the modern plant breeding, which started in 1940s and 50s. Of course, the environment is harsh. Maybe the most very continental, because we have severe winters, which doesn't allow us to grow winter wheats, and we have also soil erosion on top of that. Shortest growing period, perhaps the shortest across the world. We have to sow and harvest wheat within 100 and 105 days. If you just delay for 5-10 days, you will not be able to harvest your wheat. And the most important environmental constraint is drought, coupled with extended heat. Coming back to the point that we need good alleles, combination of good alleles. This is a nice example. Yesterday, Johan just touched about the Green Revolution, and that this is the result of the Green Revolution. The European countries mostly using those RHD genes, they doubled, sometimes tripled the yield. However, seems we just missed that opportunity out here. Even Australia and Canada, their breeding efforts focus on collecting those good alleles in combination. Why I'm talking about Australia and Canada? Because they are very similar in environment. They have very similar environmental conditions like Kazakhstan. Even they have shifted to shorter varieties, of course, in combination of these most important alleles like PPD, VRN, and the other RHD genes. This is a paper saying although we haven't changed, there was no change in plant stature. There was a yield increase to around 0.6% per year in last century, 100 years. Although there is no there is no correlation between yield and the plant height, which we again approved in our paper using that mapping population. The same authors hypothesized at the end of this paper, saying that further yield achievement should be achieved by reducing or changing the plant stature. So what we have done, Of course, we crossed the Pamitiaziva, which is the variety with Russian origin, but well adapted in Kazakhstan and registered as a commercial variety. We crossed that variety with Paragon because in order to have access to those alleles, we do need a cross variety genotypes that are genetically a little bit far. And we created mapping population recombinant inbred lines, and we identified several QTL that are associated with yield, adaptation, and with their components. But it doesn't mean that we can use those genomic regions directly into our breeding scheme. First, we have to validate. Among these QTL, the plant height controlling QTL on wheat 5A and 6A chromosome was a kind of source for my PhD. Why? Because they have a impressive, very large additive effect. Each QTL adds to the real plant height about 9 and 10 centimeters. During my PhD with the well-established near isogenic development protocol in Simon Griffiths' group, we created a near isogenic lines for those two QTL. As you can see, the difference between the near isogenic lines were significantly, statistically significant in a controlled environment. Of course, next stage is testing the effect in real field conditions. And we conducted a large-scale field trials in the UK and the main wheat growing areas in Kazakhstan, such as north and south. In the north, we grow mainly spring wheat. In the south, we grow mainly winter wheat. But however, the main wheat growing area is north, about 11 million hectares. So something interesting happened. So in the UK and the Kazakhstan 6A increased plant height. However, 5A QTL silenced in Kazakhstan were it bred for. However, it's always stable in controlled environment, also in the UK's environment. Then that was the question. Then we addressed later. Now I'm showing the yield results. Of course, we do have those alleles, height increasing alleles. The question was why we are keeping them. When I talk to breeders, they don't. No one could answer me why we are keeping those alleles, because they don't do molecular analysis and that they don't know what genes are controlling their plant height. As you can see, large-scale field trial across the years in Alm, which means in south, Pet, 3 years in the north, Kazakhstan, Petropavlovsk, we didn't see any yield advantage coming from these yield height increasing alleles. Instead, there was a single case in Alm, ALM 2021, the yield of the 5A plus, which means the plant carrying that height increasing allele, yield was significantly lower than the short. Of course, then we thought perhaps they do need to be together in order to show their full potential. And using QTL parameting method, we collected those two alleles in Paragon's background, which is the UK variety, as I said before. And as you can see, plant height of the Paragon increased by 20 centimeters, exactly as expected, 10 centimeters from each gene, each QTL. So just for the sake of understanding, these near isogenic lines, they are like Apple phones. Small tweak makes them very different, but at the same time, very identical. And that they are like twins, genetically very similar, up to 90%, but they have a huge difference in phenotype, in this case, plant height. So by the way, I'm not advertising any Apple product. It's just for the sake of understanding. So then when we generated this, we called them double nils because they carry both height increasing alleles. We tested them in the UK. As you can see on the left, A, the Paragon's plant height was almost as tall as the double nail, Pamitia ziva, sorry. So about 1 meter 20 centimeters in the UK environment. As expected, yield was negatively correlated with plant height in the UK environment. In Kazakhstan, in the north and the south, we tested them as well, double nails. As expected, the double nail was shorter by around 8 and 10 centimeter compared to the Pamitia ziva, which means even though they are combined, 5A QTL is still silenced. And again, we did find that there is no clear correlation between yield and plant height. Another interesting fact we found, the Paragon, even though it was almost tall as Pamitia ziva in the UK environment, it didn't lodge. Comparatively, Pamitia ziva in the UK environment, it has lodged around 35%. So it could be a nice source for the local breeders in order to increase the lodging resistance, because lodging still be the main problem in North Kazakhstan, which is the main wheat growing area. Of course, any PhD student wants to find the gene. It's not easy, as Brenda said. We didn't use, the deep and sophisticated genomic bioinformatics analysis, but we were able to squeeze the region significantly from the initial QTL region. For example, 6A QTL, when we found, it was 530 megabase, which consisted of around 2,500 genes. This is due to perhaps the limited recombination rate in 6A QTL of wheat, which was reported by many papers. And we resequenced the parental genotypes, and also sequenced the exons in order to develop a kind of co-segregating diagnostic markers to generate a recombinance. So then we tested those recombinants in environment, in the UK environment and controlled environment. Through integrating them, we were able to squeeze the region from 530 to 28 megabase region, which is still significant. We have done same thing for the 5A QTL. Initially, it was 37 megabase, which consisted of around 500 genes. Now we squeeze the region to 19, but still have a lot of genes, 170 genes. Then we were able further to squeeze the region to 12.5, then 1.2 megabase, using the diversity panels, which I'll be talking in my next coming slides. So using in silico available data, genomic data, even from these 180 genes, 21 were somehow related to plant height. There is a Caubin and Jediflux. Caubin means Central Asian wheat breeding panel, which consisted of around 540 genes, which I'll be talking in my second slide afternoon talk. Jediflux is the Western countries' wheat breeding panel, also around 500 lines. We used our diagnostic markers and tried to squeeze the region. As you can see, we get some HTB, which means the haplotype blocks or haplotype groups. Based on that haplotype groups and single marker effect, we were able to squeeze that region into 12, as I said before. Then further, we were able to squeeze that region 1.2 megabase. Why? How? Because we combined the data from 6A and the 5A, because we thought, okay, this could be, the effect could be due to 5A in case of 6A or vice versa. When we combined, we found kind of possible recombination breakpoints. The plus means poles, minus means shorts, blue means the height increasing area, coral means short allele area. So then there was a haplotype block 11, although it contained a oral bed, it was still tall. When we look at these population structure, it consisted of very old varieties in Jadiflux, which were released in 1940s and 50s. Same thing was done for 6A QTL. Interestingly, we expected Calvin to be fully fixed for this blue bit because this is the Central Asian panel. However, we expected Jediflux being like segregating like 5A QTL. However, we only found 2 haplotype blocks in both cases. Now it could be due to that limited recombination rate in 6A. But it doesn't mean that European wheat still tall, because they integressed other alleles that keeps their varieties short. Within that squeezed region, we have done some kind of in silico functional gene analysis. There are some promising genes, for example, for 6A, RLCK85, which is highly expressed in peduncle length. In wheat, in rice, sorry, the overexpression of these gene in rice shortened the peduncle length of rice. Why I'm saying this? Because during my PhD, we also very interested where these genes that drive the plant height is expressed in plant's organ, and we mapped the effect of 6A QTL into internode 1 and internode 3. The effect of 5A was mapped to internode 2 with overlapping effect on internode 2. So this is very coincides with that hypothesis. For example, for 5A, AXR1 gene is expressed in peduncle 2. So this is the map of all genes that wheat 6A contains and wheat 6A and the 5A contains. If you look at the 6A chromosome, most of the plant height controlling genes identified in wheat located on 6A. Our gene on top. So we really expect this gene to be a allelic version of RHD25 or RHD25 itself. However, 5A QTL could be a new gene because on 5A is only 12 RHD12 is available, and the location of our gene is far from the RHD12. So this is a conclusion, which I may just read it down. The tallness of Central Asian wheat is controlled by 2 major QTLs, which have large additive effects. The production of NILs showed us that the height increasing alleles confer no yield benefit, and therefore it could be a interesting genetically, also practically breeding back, which means decreasing the height of the Pamitia ziva with the short alleles coming from UK wheat paragon. So with this, I'd like to thank my supervisory team at that time and for your attention. Thank you very much. I'm happy to take any questions.
Yeah, thank you, Kenneth. It's really interesting. Are there any questions? Solomon?
Qanat, thank you very much for your interesting presentation, your work. So my question is belonging to the mapping populations. Just you use the biparental mapping population, yeah? What do you think about which mapping populations are very suitable for quantitative genetics? For example, yes, we know that multiparental is good, but maybe which one? NAM or a magic population on naturally natural populations, what do you think?
Yeah, of course, if you have magic population or non-population, there is a great resolution compared to the reals because you get a kind of large mosaic genome compared to recombinant inbred lines. But in our case, the task was very simple to identify the genetic regions for plant height. So most people use recombinant inbred lines for many purposes. And when you compare with double haploids, the recombination rate in reals is higher than the double haploids. And the problem with the recombinant inbred lines was the generation time, which could take the up to 8 years if you don't have any controlled environment. But this problem is now solved by speed breeding, as Brad mentioned.
My next question, you said that there is no correlation between plant height and yield, yeah? But how I know a lot of references highlighted that there is a correlation between the yield and the plant height. How can you?
Sorry, you mean plant height being correlated positively with yield when it is tall or short? For example, in Europe, it's negatively correlated. When it's shorter, there is an optimum plant height. Of course, it shouldn't be too short and too tall. And they found that this is an optimum plant height, and this plant height is the very good plant height for good yield. In our case, mostly we keep very tall varieties like landrace like varieties for feeding purposes. They say we grow tall wheats because we have old combines.
I mean, how the plant height is scored, and then the grain number, grain size might be increased.
Sorry, Azad, can you repeat the question?
So how the plant height is short, the yield might be high.
Oh, okay. So as I said, during Green Revolution, just scientists identified RHT genes, and that they are kind of insensitive to exogenous applications like gibberellin. When you just put the fertilizers, when you use that alleles and put the fertilizer, it doesn't grow tall. What it does, they directly through all nutrients to the spike rather than other plant organs like the stem and the leaves. Did I answer your question?
Thank you. Good.
Yeah, thanks for this very interesting talk. You might be aware, working in this topic, that plant height is also associated with the length of the coleoptile. And there is a certain limit, and it was observed that this shortest cultivars may have a problem under stress conditions where a longer coleoptile is required. Do you consider that in your collection, in your selection? So I actually would always have a look at that with all accessions. And as far as I remember, this correlation is not that strong in genes which do not directly interfere with the gibberellic acid biosynthesis and perception. And you showed one candidate gene interfering with BUG1, which is kind of interacting with BRI1. And I would consider that one of the most important, one most interesting
it's a very interesting candidate genes. We are just keeping there. There is a list that contains a lot of genes, but I'm just showing them as a most promising candidates. If you could offer any ways of these cloning these genes, I'll be happy to chat with you after or anytime.
Thank you.
Boshqa savollar bormi? If not the case, we in our program little change because actually yesterday we had already too much talks. That's why we skipped one talk. So maybe we finish a little bit earlier than expected. We have still time for to go to lunch. So now I would like to kindly invite Dr. Ayşe Nuran. She will be her presentation is about innovation and genome editing for crop improvement. Stage is yours, please.
Yes, I don't expect my presence presentation is today. So I am Dr. Ayşe Nuran Çil. I come from Turkey. For a long time, I manage modern plant breeding and accelerating centers as a chief of my duty. So I am sunflower breeder and peanut breeder, and I study molecular studies. So our topic is classical and biotechnological breeding methods in sunflower. Eastern Mediterranean Agricultural Research Institute project applications. Our aim every time in sunflower breeding, traditional breeding and combining the modern techniques. So maybe our presentation different to the others because I didn't give one project or one result. Only our institute's projects we introduce and we discuss our projects and techniques. Traditional selection and crossbreeding process because I study relationship between different sunflower types, oil, confectionery, and ornamental and characters. Modern breeding methods olarak tissue culture techniques, transgenic plant techniques through gen transformation, mutation breeding, marker assistant selection, plant bioinformatics and gen editing technology, gen sequencing, expression analysis, gen meanings, connections and interactions between gens, gen silencing and regulation. It is newly work for ours. Techniques for shortening the breeding period in sunflower, classical, modern, and biotechnological methods. Yes. Traditional selection and crossbreeding, everybody know. Percentage of 95 cross-pollinated plants, sunflower. The selection of the parents to be cross-pollinated, sterilization, dranking, cross-pollination, and crossbreeding techniques are crucial. The general goal of cross-pollinated plant selection methods, such as recurrent selection, is to move the genetic and trait characteristics of population. However, in the development of inbred lines, selection of poor lines is used to develop a single plant line, not a population. Similarly, backcrossing allows for the transfer of single can traits such as disease resistance to a plant line. When selecting parents, it's crucial to ensure they process traits appropriate for the sunflower type to be used. The poorer selected parents, the easier it will be predict and determine the traits of the resulting individuals. Furthermore, selecting distantly related parents will be broadened again. For example, they will create resultantly high equality plants with a wider range of traits. When hybridization sunflowers, a pollinator plant with the desired traits, is selected as the father, if the plant has been selected through positive selection, either by manually removing the male organ, we make emasculation, or by using ectoplasmic male sterile CMS lines as the mother plant pollinator. Breeding objectives is sunflower include developing varieties and producing hybrids. Hybrids emasculation techniques can be used for both. But CMS line is generally used in hybrid production. Male sterile lines are obtained using chemicals such as gibberellic acid and ethidium bromide through spontaneous mutation in nature, through interspecific crossbreeding, or through genetic engineering. This is clear, that table. The goal of plant selection based on traits is to improve or refine the resulting plant population structure in terms of the desired trait, while maintaining the plant's genetic makeup. Thus, by selecting plants with the desired trait, the plant population will gradually become poorer in terms of that trait. Generally, a high heritability of the selected trait is desired. Heritability represents the amount of genetic variance within the genetic variance. Therefore, traits with a high heritability are less likely to be affected environmental factors. First of all, we use CMS lines, and we want improving mother lines. We use resources of CMS, and then we hybridizate maintainer lines. So after 5 backcrossing time, it is ready for as a mother for hybrid as a hybrid. So we added hybrid restorer, and then it is hybrid F1 variety used for farmers. If the heritability is high, genetic process occurs rapidly. Genetic process is defined as the positive differences between a population and the previous generation in terms of desired traits. Correlative traits, those controlled by a single or a few genes, have high heritability, resulting in rapid genetic process between the population. for that trait. However, quantitative traits controlled by many gens for multiple gen groups have low heritability and experience slower genetic process. Traditional selection, crossbreeding, like this table, which year is we need? If we want homozygous inbred lines, 7 or 60 times is needs in traditional. But it is consume a lot of time because 15 years needs for F1 seeds. So first you make backcrossing. It's need time 9 years, but restorer lines, if you improving, 6 or 7 years needed, and then you make hybridized and going to go to this inbred lines ecological test. So many locations. So it is 15 years, sometimes not enough, but we want to, our aim, classical breeding F, and we use modern techniques, we shortening the breeding cycle. All of study, we conduct it. Also, we breeding aims we choosing how to be choose and selected. If it is oil or confectionery or morphological and physical characters, every trace we choose in 5 conditions. Yes, you see some varieties, but I want to say how to be shortening the breeding cycle. It is important for us. Classically, we make crossing after in our studies for first generation in 5 condition. Embryo age is 14 days. 14 days we immature embryo we pick up and then inoculate especially MS media. So you see in the picture which time we cutting the heats and put on the refrigerator. After these seeds separate, and then you can see root is very important. In this step, yes, we determine how maybe our aim is down the middle of improving. That gene we choose. But after that, it's a laboratory selection. If the root is good, like this picture, it is very useful for acclimatization, like this picture. This is a good, and maybe third root length is maybe 4 centimeter is enough for us. Firstly, we initiating this study, normal 8 or 10,000 that under the white light is enough. But when we pick up the embryo, we put on the media, and this time transfer the pod maybe 35 times fast. But you can see if we put on the under the LED light, this is at 15 days. So getting the generation, first of all, 4 or 5 times. This is if embryo improving under the light light, it's 6 times a year. It's a very important sunflower breeding. And it is useful techniques because after the pods, you transfer it fired, and then you selected it, okay. And again, pick up the embryo and you continue. In my condition, we study in Adana, so 270 days, sunny days, and minus 10. So it is very good condition for sunflower breeding. So 3 times we get inbred line in 5 condition. One condition we added greenhouse, first embryo rescue techniques for 4 and 5 times in a year. If it is under the LED light, 6 times is possible. Of course, mutation breeding, there was the other techniques. Sunflower exposed to methyl sulfonate are mutated to become tolerant to post-emergence herbicide imidazolene. Furthermore, using MS sunflower lines with the high oleic acid content can be obtained. Lines with high stearic acid have been obtained through exposed to certain chemicals. Cytoplasmatically male sterile plants can be obtained from sunflower material exposed to certain doses of gamma radiation. Ultrasound treated immature sunflower embryos can be rendered resistant to orange and can be inbred into the lines with high oil content. And we use mass marker assistant selection. The last picture you can see in pot, the sunflower, it's very small. We make classical breeding, hybridization, and then we use immature embryos from embryo rescue technique. Nanos. Oh yeah. Ben geri alamıyorum. Yes. This time, we study mass selection for downy mildew. We applying this stage downy mildew. So we choose marker assisted selection the same time. It is resistance or not, we could understand and transfer it to classical program. Yes. In this project, sensitive lines and PL13 gene resistance lines make hybridization, and then we choosing in capillary electrophoresis system and the others variety. And sometimes we use agarose gel, but sometimes we use capillary electrophoresis systems. The other marker assistance work is reagents. It is very important for us. But we say we want to shorten the breeding cycle. The others refuges, because refugens is very important for us. A male, female lines improving classical via classical breeding. It is take a long time. 9 years is we need. So all homozygous nucleuses we choosing some markers. Here. First, our study molecular characterization we need. We use many marker techniques, ISSR, SSR, and sometimes retransposon gene. We select it. It is we characterize molecular characterization and molecular characterization. So it is also how we hybridize inbred lines. We can understand easily. Yes, it is our work. And then the other project is determination of downy midwife races present in Turkey. Firstly, we collect all the like the picture, first picture you can see, downy mildew. It is an obligate parasite, and then we all collected, pick up the leaves, and then we protect its minus 80, and then we sometimes make solution, and how many sporangium we wants to make, we are calculate under the microscope, and then our inbred lines we make downy mildew tests. You say you can see it is be sowing pot, and then it is not green. It is not green. So we planning, and then which rice we want to test it. That rice we prepare solution, and then 2 days minus 60 for 4 hours we are put it on minus 16 in the laboratory condition. After that we are sowing pots again. After 2 days, we see all the leaves white sporangium. So this is classical determine the W mildew of rice. This is a technique by Thornville techniques. This is determined classically. Every verse can use this technique. So we determine again in molecular via molecular techniques, and it is which W mildew sporangium obtaining is very difficult. So we determine which isolation techniques. It's for it is very difficult for us, but we determine which isolation techniques we can use, and then which primers we can use. They are detected W mildew. Yes, and then we sent sequencing after the first PCR. We can see all the bonds, and then we can send sequencing. So it is we can combining W mildew tests. Which rice is inbred lines includes or not. We determine classically methods and molecular methods. And then I mentioned about first explanation. After 8 or 10 days, all embryos we pick up this procedure completed if we put the embryos culture under the late light. We know which waves and intensity we know. So it is possible to get 5 or 6 generation a year. So our condition in Adana, I say it is a sunny day. We have natural conditions, 1 or 2, added greenhouse 2 and 3, embryo culture greenhouse 4 and 5. If we add it in under the late light condition, it is possible to get 6. Yes, we initiating some protoplast studies. Some of them we studied. First plants were grown under the in vitro condition. It is a cut into the small strips and left in the enzyme solution overnight. It was centrifuged and started in the protocol counted on a hemomatometer. Them at 1988 and Sankdong 2013. Approximately cells were obtained. Yes, light microscope maybe Arabidopsis, but it is useful techniques, and we added electrofusion techniques. But I want to show you obtaining colors from sunflower leaves. The leaves obtained from sunflower seeds grown in the field were the first sterilized. It was when cut into the small space pieces and placed in the previously prepared callus obtaining medium. The prepared petri dishes were incubated in the dark and callus formation was observed after the 2 weeks. But we cut the study. We didn't get any goodness, so maybe we continue this study. But our project drought resistance line and variety development expressions. It is very beneficial. We use qualified study for us in real-time conditions. Helianthus annus type 7 expression is conduct successfully. So you see many parental lines and our hybrid lines, very differences. So we aim to, we have two techniques, 2 projects about drought. This is a real-time condition in our laboratory. We determine two markers give a good result for us because of the sequencing is very expensive. If we use these techniques, we wonder, but it is successful. But all the morphological monitoring, physiological monitoring, and biochemical analysis we can do. Also, we measured in chlorophyllmeter and thermometer. All of conditions we use this, and then also we we conducted in percentage of resistance restricted irrigation we give. We make 3 replicates every direct line, and then irrigation is percentage of 33, irrigation of percentage of 66, and all percentage of 100 we conduct, and it is we pick up their leaves only first of all the flowering time, because the consuming restrictive irrigation is very effective sunflower. The yield is half of. It is very important stage. So we can take RNA, and make isolation, and we conduct in real-time PCR. So we determine Helianthus annus type 7 and Helianthus annus DHN1 gene. So it is easily and cheap way for us. The other drought project is an expensive project. We all morphological, all biochemical conditions all proline and monohydrate measured. Everything is good, but different different lines. Only parental lines is to project. We use only parental lines. Yes, you can see this is not turgor because irrigation is 100 is okay, but irrigation not limited, but percent this is 5 kilograms soil there was in, and then irrigation 66 is we give we calculate them for poach 5 kilograms liter su butter we give. The others irrigation is percentage of 33 2 and dot past liter we give. Everything is okay, not turgor, and this time we took leaves. Maybe we collect 4 leaves. One of them for sequencing analyzes, one of them monohydrate and proline analyze, and maybe biochemical analyzes NADPH, hydrogen peroxide. The others parameter be measured. And then RNA sequencing studies we sent. And then this not molecular characterization. This is false. SMP, we have SMP data, so some primers be improving, and we make validation studies. And then I didn't give the result, but we determined 5 genes related to the drought, and then 5 primers is validate. Like last project, we understand aquaporine gene, but this is our new primers. Maybe we use in the future not sequencing, but gen expression techniques. We will continue because it's cheap. Transgenic plant breeding through gen transfer is very important. Sunflower breeding also. Agrobacterium mediated gen transfer. This is activated by introducing the desired gen into the target cell using the bacteria, such as Agrobacterium tumefarensis and Agrobacterium rhizogens, which contain plasmids. Also, this is a new project for us. We initiating some chemicals, electrophorator, many techniques. We initiating. We don't know results. Thanks for your attention.
Yeah, thank you for your talk. Are there any questions? about sunflower transformation.
Thank you for your talk. I just did you try the transformation of sunflower for the plants? Did you try? I didn't hear. Did you try transformation of sunflower protoplast? Yes. And again? But on the slide it was Arabidopsis. Yes, yes. Be clear, it, but maybe don't sen kaydetmemişsin, kalmış yani.
Kimdedir soru var mı? Ha? Savolda. O'zbekchada. Turkchada kim ayta oladi shu savolni? Ayting. Sunflowerni o'sha protoplastini qilganman.
Protoplast işi şimdi var mı burada? Project işinin içinde. Project içinde protoplast işi var mı?
Evet, var. Var ama
Oraya.
paradox yani I express my presentation about sunflower. So she write say you see Arabidopsis. So yani silmeyi unutmuşuz.
So'rlar bormi boshqalarda? Thanks so much for your talk, but I'm sorry for the inconvenience because you supposed to be give talk yesterday, but because of the arrangement, you postponed today or tomorrow. But anyway, thank you. And our session going to finish, and it was truly engaging. And please join me giving big hands to all our speakers. And then now we have a lunch break, and